on experimental animal handling and environmental enrichment. I am Dr. Poonam Daugule. It is my privilege to welcome you all as an anchor today's academic sessions. The day one was highly informative and enriching, and we have confidence that day two will be equally insightful, engaging, and rewarding for all our participants or delegates. So we move to the first session. The day two first session. For this session, we are honored to have with us a sincere personality, Dr. Neeta Burande ma'am. And this session will be chaired by Dr. Ashwini Kale ma'am from CIR department. I request to Dr. Neeta Burande ma'am, please come on the dais. Now I request Dr. Yashwant sir to kindly introduce today's eminent speaker, respected Dr. Meeta Burande ma'am. Please sir.
Good morning, respected dignitaries, colleagues and students. It's my honor to introduce Dr. Neeta Burande ma'am, an accomplished academician, clinician and researcher with over 2 decades of dedicated service in pharmacology and diabetes care. Dr. Neeta Burande ma'am completed her MBBS from Government Medical College, Miraj, and her MD in pharmacology from Dr. VM Medical College, Solapur, where she stood first in Shivaji University and was awarded with Jezukar Prize. Her commitment to diabetes care is reflected in advanced certifications from globally recognized bodies including the University of Newcastle, Australia, the American Diabetes Association, and a fellowship in diabetes endorsed by the Royal College, United Kingdom. She has an impressive 14 years of experience as a professor and head of Department Pharmacology at D.Y. Patel Medical College, Kolhapur, and continues to serve as a professor since 2022. Along this side, academics, since 2018 she has been actively working as a diabetologist, researcher and administrative director at Surya Hospital, Kolhapur, and integrating clinical care with research and education. Dr. Burande ma'am has made significant contributions to medical research with more than 25 publications in reputed journals, along with several abstracts and book chapters. Her research focus mainly on diabetes, food infection, pharmacovigilance, rational drug use, biomechanics and clinical outcomes. She has presented her research at international forums in Denmark and Turkey, and at major Indian platforms including DFCCON, International Diabetes Summit, and MPS National Clinical Trial Summit, where she also served as a speaker on informed consent and ethical clinical research. Beyond publications and presentations, she is formally trained in bioethics, medical education, technology, competency-based medical education, and holds an International Child Act Fellowship in Clinical Research from the University of Malta. Now I request Dr. Neeta Burande ma'am to begin the valuable session and enlighten us with knowledge, sharing us valuable thoughts and words. Thank you.
Good morning all. The topic is CCCSE guideline, and we know that there is a well-known website for the guidelines as well as act and rules. They have almost more than 15 PDF for all different guidelines which can be downloaded freely, and my today's talk is wholly based on that. So what we are going to discuss today is the CCSA acts, rules and guidelines. So there is there was the first act by the government in 1960. I mean this one. So the act was by the government in 1960 about the protecting the animals through law and compensation. So in which if you see the chapter, there are the 6 chapters having the comprehensive framework that in 1960 it has been amended many times. It is applicable to whole India except J&K, and animal welfare is mainly for the prevention of the cruelty. So it's not work. ठीक, I will try once. So there are few definitions like what do you mean by animal, any living creature, domestic animal which is tamed, captive animals which is kept in the confinement, and owner is the any person who is in possession or the custody. So the fundamental duty is every person having care or charge of any animal must take reasonable measures for the well-being, prevent unnecessary pain or suffering for the animal, and exercise proper supervision. So the chapter 2, it defines mainly the establishment, constitution and terms, which is body, officials and the constitution every 3 years. Chapter 3 of the Act about the board functions like legislative review, what are the laws, what do you mean by the veterinary care, how should be the shelters for the animals, how we are supposed to transport the animals, how we are, what should be the slaughterhouse design, and along with that the public education. Chapter 3, they also defined about what are the prohibited acts of the cruelty. What should not be done. For example, physical abuse, overwork of the animals. If they are unfit for the employment, then they should not be. It is all about animals. Harmful substances should not be given. Transport should be comfortable. Confinement should not be crowded. It should be better with adequate case sizes. There should not be neglect. They should be provided the basic food, water, light, shelter, and they should not be left alone to suffer. And definitely the other methods like mutilation or sterilization should be done with the justice. So all these things is defined in the chapter 3 of the Act. Chapter 4, it gives you about the guideline about how to do experiments on the animal. So experiment on the animal, they have defined the purpose, what are the permitted purpose, are the advancement of the knowledge, saving the lives, alleviating suffering and combating the disease for the human. Then the control, they have established the committee, that is for the committee for control and supervision, and the power of the committee has been defined like inspection, rules, prohibition as well as penalties. Now, the safeguards for conducting the experiments, there are some pillars. The pillars are like registration is needed, then you need anesthesia when you are doing the experiment on the animals, the destruction should be humane, alternatives, prefer alternatives whenever they are possible rather than using the animals. Prefer smaller animals whenever possible and then go for the larger in the hierarchy. And definitely you have to take care of the animal before the experiment as well as after the experiment. Chapter 5 of the Act, it defines about how you are performing the offences, so what are the penalties or what should not be done. So chapter 5 mainly is about the penalty and then enforcement. So in which they have given the power and unregistered exhibition, unauthorized animals, obstruction of the animals and concealment. Sometimes you have the animal and you just don't disclose. So all these things are prohibited and they can be attracting to the penalties. Chapter 6, again the extension of the same, that is enforcement. So if you see that the committee or the people from the government authorities are enforced. They can come and they can search the animal house. Or suppose if someone is doubtful or something, they just can come directly to the animal house. They can search and they can say search. So for that, warrants may be required. Sometimes there may be surprise inspection when you are having the register. All the animals can be examined by the veterinary purpose. Then the powers given by the court to them are they can forfeit them, they can custody, they can remove the custody from you and they can take the animals with them and they can keep them for the shelter or for the modification in the animal welfare house. So definitely all these facilities and power are given to the government authority. Treatment and care facilities, like when they have to send the animal, then there are the different facilities like infirmaries, pinjra poles, and fitness centers and the certification by the government. So these are the acts which has been given in 1960. After that, there are the rules. There are the rules based on that act was given by the government and has been amended many times. Like so the rule was first drafted in 98, then it has been amended in 2001 and then it has been amended in 2006. So let's see how it happened. I will go chronologically what was first and how it has been improved or modified. So when the rule has been formed, they have given the structure of the committee first. They have formed the committee and given the structure. So they have formed two committees. One was the CPCSEA, which was the older one. Now we say CCSEA, Institutional Animal Ethical Committee. So one is the Central Committee and one is the Regional Committee or the Peripheral Committee, which is attached with the institutions. Like our own hospital, our own college also have the IAC. Then they have defined the definition. So breeder, the person who is breeding, establishment, where the committee is like, and experiment which is governed done by the usually the researchers or the PG students. Registration, mandatory, all these three has to be registered with the CPCSE. Application process is simple. You submit the application, PMI is verified and registration is given. Now this verification of the premises and the member, it is the where the guidelines work that we will discuss later. Then there are some basic requirements. It is the rule. By rule you have to keep all these things. So for example in the documentation. So you have to keep the mandatory register about the animal, health status, experiment and reasons why you are using that particular species for the experiment. So these are the mandatory documentation which can be checked by the members. Then they have given you the rules about the animal welfare, like location, it should be undisturbed, cages should be not crowded, trained staff. There are the training which are available. Like I think in Hyderabad, in NIN also there is the animal house attendants and cleaning. Our animal house attendant is trained from there to take care of the animal. And then care should be continuous. So for the permission, suppose if you want to do an application, it would be scrutiny and there would be the permission. Now the next question was there, like okay fine, permission is given, but who can do the experiment? So that time they have made the classification like anyone who has the degree or diploma in veterinary science, medicine or the laboratory animal science from the government recognized university. That ethical standards who have been maintained like care, proper care and supervision. Then comes the question of the anesthesia requirement. What anesthesia should be given? So mandatory, previously what, I think when I was a undergraduate, that time there was something like stunning the animal or keeping them without the anesthesia or doing the experiment without the anesthesia. It was not streamlined. So they have done the guideline mandatory to anesthesia even how short the experiment is. If it is painful, you have to relieve the pain. So this is first thing. So this was the, so what they have done, The anesthesia has become streamlined, administration has been become streamlined, and destruction of animal has become human. Then practices which have been prohibited since then, no public demonstration. You know, like जो पुराने लोग हैं उनको पता होगा वो बैल का रनिंग होता था या बहुत सारी जत्रा वगैरह में होता था पुराने इसमें. So all these things have been prohibited. Nothing can be done. No illustration. Suppose if I am supposed to teach you and I am saying, okay, I will show you how to cut the abdomen, how to show the frog heart or rat heart. Nothing can be done for the illustration purpose. If it is doing something which is not available, it is the part of the research, and that time if you are demonstrating, then it's okay. Someone is looking and you are doing already, which is giving some knowledge, new knowledge, then it's okay. No skill training until there is the recognized training institute, and no repetition. You cannot repeat the things again and again. Then there were the rules for the transport in which they have given. So that was a very important rule that you can't just take like अगर अभी मुझे खरगोश ससा. So if I want this rabbit and there is someone who has the rabbit and I will say okay I will purchase it and bring it in one animal. Not it. It has to be like breeder should be registered as well as establishment should be registered. So when both of them are registered, then only transfer is allowed. How comfortably you are transferring is not important. That is also important. But first requirement is both should be registered. So it should be registered. Establishment should be registered. No import if animals are available domestically. Like you cannot import from outside the country if they are available. Suppose if the animal is available, like for us example, if the animal is available in Bharti or some nearby, then there is no need to take the animal from Mumbai. So try to procure as near as possible to reduce the transfer stress or the transport stress. Okay, we can produce the animals, we can breed the animal if it is permitted by the standard company. Like our establishment, they are, I think our animal house is allowed to breed of the certain number to maintain the number. But we cannot go with the genetic modification. For that again you need the separate guideline. There was no contract research initially. No contract research is like suppose I am the company. I am a pharmaceutical company and I am saying okay this is my drug and you do the research. It was not allowed previously. Amendment they have been allowing it with some specific purpose. But initially it was not allowed. Yeah, but collaborative research is allowed. There are two academic institutions and they are doing the collaborative research. Then the contract, it's not contract but it is the collaborative research. So this is how it has to work. Then there was the composition which they have defined in the IAC. What should be, it is for the institute level or the establishment level. There should be one biological scientist, two scientists from the different discipline, one veterinarian, one facility scientist who is the animal house in charge, one external scientist from outside, and there should be one public aware or the public representation, that is the non-scientific member which is socially aware. So usually the MSW or the lawyer or anyone who is working in the NGO can be taken for that purpose. Sometimes your project is so specific that probably you have to add one or two co-opted person for that research specifically. For example, like now in our college, nanoscience or the nanoparticle research is very common. So in that, if you have taken a person, among these no one is expert in nanoscience, then you can co-opt one more person who is expert in that and who can review your protocol. Okay? So this is about the committee. Enforcement, again we have said that whatever the reports is received by the committee, if there is a non-compliance, then they enforce the suggestion of the experiment. So it is why I have taken this slide, that don't feel that if you have got, they are finding that it is not followed, they can seize your practical. Okay, so it is important that you have to compliance after the experiment also. Then you have to keep the documents along with you for 3 to 10 years. So that we will see. Now there was the amendment. I have told you like there was the rule which has been formed in 1998, and then the amendment was the expanded definition. Experiment has included the production, testing and transgenic. You see how the development occurs in last 98 to 2001, almost 12 years. So transgenic animals were in the streamline for the experiment. So they have expanded the definition. Record keeping has been mandated previously also, but now here it is daily. You have to put the daily recording. It's not like end of the month, जो भी पिछले 30 दिन में हुआ वो मैंने लिख के खत्म किया. It should not be like that. Because if I am coming today, I will check what you have written today. तो आप ये नहीं बोल सकते कि हाँ 10 दिन पहले तक का तो लिखा हुआ है. It's not like. So whatever you have written for 5 years is all in vain. So daily register maintaining is very important. Qualification, they have expanded the qualification properly, like I will show you in next slides. Timeline clarity. पहले कैसे था application दिया 6 महीने नहीं पता चल रहा और you are just waiting, they will answer. So they have made the timeline that if I have given some application, some query or some new submission, then it has to be answered and should be completed within 3 months. Housing standards, they have been defined. Like if you see in the rules, it was like it should not be overcrowded, it should be comfortable and all these things. So they have made it clear. Like how what should be the area, what should be the ventilation, what should be the day and night cycle, what should be the temperature. So they have been specified it. And along the other provision which we have done before, they also prohibited the paralyzing the animal. Like you have given the QRAM, the skeletal muscle relaxant. So a person that animal is not moving, but that death is really very painful because your all brain is active. So the skeletal muscle relaxant without respiratory support is the hell, the worst experience for the animal. So it is totally prohibited. Okay. Again, the definition already we have discussed. These are the different definition which has been expanded. In the registration, compliance and timelines already we have discussed. Process already we have discussed. Record keeping, I have again to review because it is very important, so you maintain daily. And what you are supposed to write in that, number, species, age, gender, and whatever the particulars, like enrolled in that experiment, given there, coming back, experiment done on that day, everything you have to show to the committee for the review. In maintenance housing, already we have discussed that guidelines have been, see these guidelines I am not discussing because they are different as per the species. Okay, so I will just go through. If you want details, you can later contact me or you can just download it from the website or I will give you the PDF also. This was more important, what are the qualification requirements. So previously it was like degree and diploma holder in medicine, lab or veterinary. Now they have explained that it should be specifically in this postgraduate in life sciences or degree in pharmacy, laboratory and medical profession. Restrictions was there already. So what they have done in restriction, previously it was completely restricted. Here they have opened it. So what they have done, school, colleges where it is for education can be done with permission. Okay, so there our establishment is where we can perform, we can show them. Paralyzing isn't definitely not there. Here what they have done, not just the DTC, not just the curate, but all paralyzing agents, all skeletal muscle relaxants cannot be given. Now acquisition rules, source has been restricted, permission has to be given, import has been limited. So this is already discussed there. Now there was a change in the contract research regulation. Previously it was not done at all. Now it is permitted. But how it is permitted? After the committee approval. You have to put the application to the committee. If they are approving, then you can. So for that you have to put it in the registration that contract research is also in the aim or the scope of the research in the institute. Academic definitely is the in which collaborative can be done. So what is the key distinction? Commercial contracts you have to take the permission. Academic collaboration no restriction. IAC approval and it would be over. But if you are doing contract, you have to take the permission separately from the CCSE. Okay. So there were the already we have done. So now the next amendment was in the 2006. The latest amendment in the rules. So in 2006 again they have expanded it further and they have taken transgenic stocks and routine diagnostics. Phylogenetic priority. Here first time we have started to say scale down. It was mandatory. It was in the rules. If the smaller animals are available, then don't go for the larger one. Rehabilitation become mandate. Till now, post experiment, what we were doing is destruction. Anesthesia to be given, SMR not to be given. Here, rehab is mandatory. It has to be included in the cost of research, which was not done before the 2006. So here, rehab has been introduced. Euthanasia has been cleared when the end of life is permitted. Previously what happened, once the experiment is there, to cut down the cost, we used to go for the end of life. But no, if animal is there, you rehab rather than going for the terminating the life. So these are the few important ones in the 2006, specifically the rehab, euthanasia and the scale down. So what was the guidelines? Paralysis and dysfunction. If the animal is paralyzed, not performing, incapable and in severe pain, then you can give the euthanasia, that is the end of life. If animal is having severe pain, recurring pain, or if the life-threatening situation is there, this life-threatening situation either to human or to other animal. Like suppose if you have done the experiment with the infectious agent, and you know if the euthanasia is not given, then probably the infection may be a risk to the other person. So that time you can go for the euthanasia. Otherwise, first rehab followed by euthanasia. So this is how the suspension and enforcement, they have become more strict and they have given the, but in that previously it was just the suspension or the seizing the committee and the establishment. But now they have done the two cadre. If minor violation is there, then suspension is not there, but just the warning or the some minor incidents. So this was about the 2006. Then what we have is the 2023. In 2023, there is the formation of committee, that is CCSE. Before that, what we were doing is CPC-SEA rules and regulation. But now actually the committee is formed in 2023 where there are the 15 members, 3 experts, 2 specialists, 2 ethical members. But the principle of that committee is 3R. Replacement, reduction and refinement. These are the 3 main things. If you don't remember anything from today's lecture, it's okay. Please just remember there are actually the 4 R's. So these 3 R's at least for any animal experiment, whenever you make the protocol, ask yourself whether we can replace this species, how much minimum number is required, and how we can improve our protocol to have the minimal pain to the animal. These are the 3 basic questions. Whenever you start your protocol or the research, go with this. So core function of the committee already we have done: registration, facility approval, inspection, IEC formation, protocol review and training. For that, the committee representative, they visit to the establishment. In committee itself, in CCSEA itself, there are we have understood the different members. Each member has 1 vote. But there are some special voting provisions. So those special voting provisions in Delhi are, if any member objects, no, this animal cannot vehicle, there are the other alternative available. Then this is the veto power. Protocol would be rejected. If number is not satisfied, protocol will be rejected. Otherwise, there is the majority rule. Votes yes and no, but these are the two conditions where if the member objects, it is the veto power and it would be rejected. So that's why I have told you go for 3R, which is most important. Now, when the CCSE nominee, they have been appointed for the establishment, what are their duties? Or what do you expect from the person who is visiting your establishment? What power they have? So, if you see, they are responsible for the animal welfare, compliances, whether we are following it or not, and they have to maintain that aura of the committee, dignity. So, how to prepare? So, for that, what is expected that whatever the protocols are there, they have to mail them before in hand. So, they will read it. It is not like I am giving the protocol now and you give the decision. I need time to read, review, to reference, and then only I will give. So review papers early, so at least give 1 month before. I think in our protocol it is 1 month before now. So we have to send the protocol 1 month before. They read it and they flag the things. So during the actual meeting, the discussion becomes simple. Even they can ask about the queries and more clarification by the mail before having the actual IAC meeting. So inspection, whenever there is a meeting, you may have noticed the stakeholders, there is always an animal house visit. So this animal house visit and inspection also they have to report to the central committee. There cannot be an IAC meeting without animal house visit. It has to. So they visit, apart from that, they may visit it unannounced and they focus on the feeding and water, which is more important of the animals. What they check, growth is normal, behavior is okay, disease is not there. Then they check for the standards, they enrichment, like how much is the socialization, what is the case design, whether staff is trained or not, and whether, this is one more very important one. When I was a HOD that time, it was the issue which I have to clear that time. How you do take care during holidays. Because there are the 2 days holiday, 3 days and everyone is entitled for the holiday. So there was the question, how you have taken care for that 3 days Diwali vacation? Diwali के बाद की बात है. And that time I told कि नहीं वो हमारा जो trainer है वो आता है. आप उसका दिखाइए in out. So they can ask anything. So after that now we have the जैसे आपका अपना हॉस्पिटल में आता है ना कि all emergency will be open even if there is a holiday. Likewise in the animal house also we have the duty chart. कि even if there is a holiday, the person is responsible to go to visit and to take care of the animal. So this was a very important part that time, even today. But I am telling you just the incident which happened that time. So these are the, again to summarize, 7 critical areas about house, feed, water, exercise, health, handling and companionship. Companionship is again here required a special mention. You cannot put rat with mice, rat with rabbit. There should be a separation of the species and there should be some companions nearby. आप isolated रखोगे तो मजा नहीं आएगा. So you need at least 2 rabbits, 4 or 5, 6 rats, and they have to be kept together, even if they are in the different place. So then in 2015 there was the guidelines, then there was the requirements about temperature, ventilation, humidity, lighting, then space requirement guidelines for the large animals and the small animals, and then comes the Form D. This is what we have to submit to the committee about the animal house details, about breed, contract, all these things has been given. They check about the Form C. It is declared what the actual which they do during the visit. They Form D if they are finding any discrepancy and they count it. So all these things are done when there is a committee meeting. And the 3 questions which are usually asked whenever you submit the protocol, the same 3 questions are asked. Whether animal are required, whether the study is done before or it is repetitive, or we can reduce the number. These are the only 3 questions. So what is the problem? Here we are not going for the research merit. We are going for the ethical merit. Okay. So this is the difference. For research merit, we already have the institutional research review committee or the IRC board, where whatever the protocols you are making, they are given in the research review. If they are accepted, then only you have to submit it for the IEC or IAEC. Okay, so this is how the things goes. It is prohibited, already we have discussed. So what is recommended? So recommended is thiopental sodium overdose. Okay, so this is how you can give the euthanasia. Responsibilities, we have to prepare for the meeting, inspection is there, documentation submission, compensation as per the guidelines. This is one more thing, administrative. The meeting has to be done once in a 6 months or more. Minimum once in a 6 months. If there is no meeting in a 6 months, then you have to inform to the CCSA member that there is no meeting and they will inform to the central guideline. So this is important. Constitution already we have discussed. So after the approval, you have the formation like it should be. This is one thing that you have to reconstitute the IAC every 5 years and 50% are replaced. So 50% in 5 years. These are the two numbers which you have to remember for the IAC. Requirements, they have given the guidelines. The socially aware person, he should be minimum graduate. ऐसे नहीं आपने अपना किसी भी बंदे को उठाया और बोला नहीं ये active है. There are the people who may not be graduate and socially active and very good work, but as per the guideline, they should be graduate. So these are the qualification which has been disclosed again. Composition, already we know that how it becomes. There are the additional IAC members. Apart from the establishment, there is a main nominee. Then there is a link nominee. What do you mean by the link nominee? If the main nominee is absent, then the link nominee come into the picture. Otherwise, most of the time we do conduct the meetings with the main nominee only. There may be external scientist or there may be a social worker. These are the quorum requirements. Minimum 6 members are required for making the consensus. All documentation should be completed as soon as possible. And when you are now ours is already IAC registered, but someone is going for the new IAC, then these are the requirements. You have to give the CV, consent, and other information. So this is biodata, academic qualification, and most important is the animal handling experience. This is most important to send along with the professional experience. That this is the reconstitution rule. So to summarize, you have the 50% reconstitution and 100% compliance. Then there were the some guidelines which are specifically for the other species. If you see, since 2000, most of the animals were rat, mice, guinea pig and rabbit. Dog and other animals were there, but they have been prohibited later with the descaling. And then there were the guidelines for the equine, for the poultry and for the fishes later on. So we just go through just the bird's eye view for that. So if you see for the equines, they have made the selection criteria, it should be 5 to 14 years, they should be in the 3 years. Plasmapheresis has to be done, LFT should be done. Then pre-purchase screening of the equines like horse and other ponies. So examine diagnostic tests to be done, function and essential requirements like health inspection and the person who is experienced in the stable. Quarantine protocol have been made about the 21 to 40 days quarantine, and then only you have to take them into the main stable. Options, there were the two options, previously there were not. So now what they are saying, either you can put in the individual stable or you can have the open barn system also, like the whole big field where you are putting the horses. So horse nature has to be understood ki why we have given the open barn system, because they are the herd animals. So they stay together, natural behavior, rolling in mud and dust is allowed, and they are the grazing animals. They continue grazing, their stomach is small. So all these things, when you go for the shelter, nutrition standards are given like about the concentrates, how much fiber should be there, how much water should be there. What you have to remember for the nutrition, the stomach is small, they cannot vomit, and there is the fermentation in the hind gut. So these are the three things which you, the veterinary person, take care of, and you have the herbs. And apart from that, there are the other things and the establishment like ration कितना है, parasite control है या नहीं, what are the immunization needed, what are the bleeding protocol, because ultimately it is used for the serum production. So how much blood you can take. So maximum 1% is required, blood volume limits are there, and you recognize the pain that never leave them in the pain. Almost. So this is the welfare about that, rehab. Then in 2020, they have covered more about the reuse guidelines, that you can reuse after the washout period rather than giving the euthanasia. So they have covered these large animals for the reuse, and they have made the special rules for the dog, that is, there would be only 3 years in the lab, not beyond that. Then certification is required when they cannot be reused is guideline. Then breeding and telemetry limits are guideline. Rehab responsibility, this was again made more clear, like it is the duty of the establishment. So if you are transferring, then you transfer with the documentation, otherwise it is the lifetime care. And when euthanasia is permitted. Then there are one more thing which has been added later, that is the adoption. Adoption, animal can be adopted after the research. So this pathway is for the dog, other species and productive animals. They can be adopted by the farmers, but please remember, adoption does not mean the selling out. Be clear, they cannot be sold, they cannot be re-adopted. आपने जिसको दिया उसी के पास रहेगा, उसके बाद में आगे नहीं जाएगा. Okay, so there are the pathways where the person can adopt them, but with the rules and regulation. Record keeping as usual with all. This is very important, that whatever the records you are making with the animal studies has to be kept for 10 years. Okay, so for 10 years your research document should be there. Now we have the unique animal identification. CCSEA, they give a microchip. आपने movies में देखा होगा ना, वो body में insert करते हैं. Likewise they insert the microchip in the animal and then they track the animal everywhere. So this is how they microchip and what it means. The only thing is in poultry, the anesthesia or the euthanasia is used ketamine rather than the thiopental sodium. Then you have the guidelines for the experiment on the fishes, because fishes, I think in last 5 to 7 years, the zebrafish and the other lower animals, they have become very common in the research. So they have made a guideline for them also, in which again you have to take the permission from the CPC SEA about the inspection and the registration. Here you are more concerned about the water quality, engineering standard of the aquarium, and again same thing, electrical safety, ventilation, lighting and planning. Critical water parameters are very specific for the fish, which has been defined. Nutrition and feeding has been defined, like the food size should be small, there should be 5 days feeding, and then the feeding has to be done for 3 to 5 times a day. So all these things, apart from like darkness allows zebrafish to reset, clutch size should be 70 to 300, and so all these things is defined in the fish guideline. The training, again the same, specifically the person who is trained in poultry or fish has to be taken. So these are the different guidelines for the different species. So if you see, it has been started only with the rat and then goes to the larger animals, then come to the poultry and the fish. So all these things we try to cover with the common concept that the less pain or the minimal pain to the animal, and take care of the responsibility till the animal is in the life, or till the person animal is having the life. So I think I will stop here. There are a few MCQs after the, I think, session is over. If you have any doubt, I am always call about beyond. So anytime you can reach with me. Thank you.
you so much, ma'am, for informative session on the CCC guidelines and their new amendment. Now I request to our puzzle volunteer Apeksha to start the puzzle.
Thank you so much, ma'am, for such an insightful session. We are really grateful to have you here. So, the delegates, we'll start with the question answering session. And I hope you all already know the rules. I'll give you 4 options and you have to answer one. And please raise your hand before answering, okay? So, the first question is, according to CPC-SCA guidelines, which principle must be followed while designing animal experiments to ensure ethical research? I'll repeat the question. According to CPC-SCA guidelines, which principle must be followed while designing animal experiments to ensure ethical research? And the options are A. Principle of maximum utilization, B. Principle of least cost, C. 3RS principle, replacement, reduction, refinement, and D. Principle of repetition. Do not answer, you just have to raise your hand. Okay. That's the right answer. Please come forward.
I request to speaker, please kindly hand over the gift for.
Please introduce yourself and tell the answer once again.
Myself, Rishikesh Chaudhary. I am from Chennabasheshwar Pharmacy College, Latur.
Very good. Please come on the dais. A big clap for him. Amen.
Delegates, please do not answer directly. Raise your hands first, okay? The second question is, as per CPC-SCA rules, animal experiments shall not be performed for which of the following purposes? Option A, advancement of physiological knowledge. Option B, saving or prolonging human life. Option C, public demonstration or illustration. Option D, combating disease. Yes? No. Option C. Yes, that's the right answer. Please come forward.
I am glad that you guys, like you were not sleeping but were listening. Thank you. But
Please introduce yourself and give your feedback. Tell us what did you feel, how was your experience in the workshop?
My name is Vaishnavi Munde. The workshop is full encouragement. I feel glad about knowing this in knowledge. And thank you, ma'am, for such a great knowledge. Thank you. I am from Sri Ganpati College Pharmacy, Temburni.
Please come on the dais for the gift. Thank you, ma'am. Please take your seat. Please take your seat, ma'am. Now I request to Dr. Ashwini Kale, madam, to summarize the session.
good morning.
morning, respected dignitary on the dais, Dr. Meeta Burane Madam, respected dignitary of the dais, Jarak sir, SS Patil sir, Principal Jangme sir, and all my faculty colleagues and dear students. So we are very fortunate to have early morning session. So the madam presentation which was very excellent. I'm very much thankful to you ma'am, very nice presentation and detailed on the guidelines, whatever information madam has been given, it is very much detailed. So in her presentation, madam covered the committee structure, CCSA guideline committee structure, and then mandatory documentation, rules and regulations for registration of animal house. Also, she covered the daily register we should have record and given amendment rules in 2001, 2006. And mostly important, those are 3 R's: replacement, reduction and refinement. So we assure you ma'am, all the researcher and student follow the rules and guidelines for the animal experimentation. So once again I thank madam for very insightful lecture. So I request student, if you have any question, you can approach to madam, you can ask. Yes, there is a question.
If anybody have a question, please raise your hand. Mic will be provided to you. If anybody do have a question, please raise your hand.
Ma'am, you tell about the adoption. Is adoption transferred genetically?
Guidelines are not actually clear defining the genetic or the non-genetic. Guidelines are not clear about the transgenic and non-genetic transfer. Right now, it is only for the routine breeding animals, because transgenic animals, they may impose the breed later on. So for that, it is not clear yet. But if they are non-genetic, definitely you can go for that.
Thank you, ma'am.
I have a question regarding when talking about the reduction of the animals in the experiment.
Please keep the mic near to your mouth.
Okay, so my question is about while talking about the reduction in the experiments, about the number of the animals. So what we can, what are the ways to deal with the less significant statistical analysis while dealing with such less number of animals?
Yeah, the number is defined by the statistical exercise. You can always take the help of a statistician to define the minimal number, which depends on the magnitude of the effect which you want to measure, what is the normal population parameter, standard deviation, and all these things give you like P ratio, Q ratio, then power of the study. All these define the minimal number which you can justify. Sometimes your study can justify even the 100 also, and you can justify the 10 also. It depends on your research protocol. It is not like if your justification is for 100, then it would be rejected. If it is justified, it would be acceptable. But suppose if your study requires only 5 and you have given the 10, then also it would be rejected. So it depends on the statistics. Yes.
I want to add, n is equal to 6 should be there for minimum. If you want to apply statistical analysis, if your result is significant or insignificant, minimum n should be 6 per group.
I think that is when there is no previous studies available.
Yeah,
For the
yeah,
first pilot
yeah,
study, you can
pilot,
go for the
yeah.
minimum 6. Correct. Yes.
Ma'am, I would like to ask that the smaller animal, yes, it's okay, but for larger animals, what kind of special facilities are required in animals?
Sir, there will be separate PDF for the each animal species, especially for the equines they have been given, and the bulk. So in equine there is the area which I have discussed. Larger animals is there in the guidelines. But especially for the horses, they have given the two options, like the parameter of the stables 10 by 10 should be there for each horse. And if you are taking the big ground, then you can put as per their area, like if your area is of 100 square feet, then you can put the 10 horses accordingly. So guidelines are there, not just for the area, but the light cycle, temperature, ventilation, water quality, feeding, everything's there.
Thank you so much. So please give big applause for Madam.
I request to Dr. Ashwini Kale Madam to kindly felicitate today's speaker Dr. Zita Ma'am with a bouquet and memento as a token of gratitude. Now I request to Ms. Samruddhi Ma'am to kindly felicitate the chairperson Dr. Ashwini Kale Madam. Thank you so much, madam. Now we have with us the dynamic personalities, guest of honor, Dr. S.S. Patil sir, principal of Dr. J.J. Magdum College of Pharmacy, and Dr. Ravindra Jarak sir from Bharati Vidyapeeth College of Kolhapur. I request to Dr. Jangme sir to kindly felicitate Dr. S.S. Patil sir. Please, sir. I also requested to Dr. Janme sir to kindly felicitate Dr. Ravindra Jaral sir. Please, sir.
सर माझे गुरु आहेत.
Thank you so much, sir. Now we move ahead. Day 2, session 2. For this session, we are honored to have Dr. Abhinandan Patil sir as an esteemed speaker, and this session will be chaired by my mentor, Dr. S.S. Patil sir, principal of Dr. J.J. Magdum College of Pharmacy, Jaisinghpur. Now I request Dr. Kadsi sir to kindly introduce today's speaker, Dr. Abhinandan Patil sir.
Good morning, dignitaries of the dais, the expert resource persons, and the beautiful and enthusiastic audience sitting. A very good morning to one and all. Today, we are here to invite Dr. Abhinandan Patil and Dr. Dawalshank ma'am to speak valuable information about upcoming trends in animal studies and advanced instrumentation techniques. Unfortunately, due to the health concerns, one of the resource person, Dawalshank ma'am, could not make it to this session. So this session would be carried forward by the Dr. Abhinandan Patil sir. To briefly introduce him, Dr. Abhinandan Patil is a DST Inspire Senior Fellow, a world record holder, and a cancer researcher with an exceptional interdisciplinary background. He has over 108 publications, 45 Indian and 9 international patents, and his PhD research was honored as the Startup Hero of Maharashtra under the Make in India initiative. His work strongly emphasizes innovation, translational research, and application orientation animal studies. Today I feel immensely pleasured and with great respect and honor I request Dr. Abhinandan Patil to take over the dais and give some valuable information and insights on upcoming trends in animal studies and advanced instrumentation techniques. Thank you.
Good morning to one and all. So today topic which is allocated is upcoming trends in animal studies and advanced instrumentation techniques, and accordingly we'll see the things. So I'm not going to bore you like that. So we'll have some basic interaction, and in that interaction we'll try to know what exactly the new things are coming up in the field of the research, so that it will be very useful for you people as a young mind, as a young researcher, to gain access to the new world. Because already you are familiar, after the COVID, the thinking nature of the clinicians, the researcher is totally changed. Once upon a time you used to say, to bring the new molecule into the market is usually used to take 20 to 25 years of the time tenure. But in the COVID, within the tenure of 1 year, the vaccine was made available. So it was due to the different types of the omical studies, we call it omics, like transcriptomics, then proteomics, and many omics are there, we are coming into the trend. Now before to start with the session, I just want to say, Einstein, how many of you know the work of this person? Albert Einstein. Are you familiar about him? Yes. Yes? Okay. So he used to say always that the whole of the science is nothing but the refinement of everyday thinking. That means whatever you think, if you bring into the concept, into the inception, then and then the things will be fruitful. So by taking his concern, we'll go to the part. Okay, life comes with options. Okay, life comes with options. Suppose if you get an opportunity to purchase the car, so what will be your liability? I just want the answer from you all people. Yes? Yeah? Not audible. Okay, new car. And suppose if you are planning for some houses. Why? Why new? Generally people say old is gold. I think last day the cost of the gold and silver rate is crashed, if I'm not wrong. Okay. Just tell me, why new? Yes, why new? What is the reason? Suppose if you get an opportunity to visit some abroad and you have the opportunity to purchase some mobiles, like which mobile you prefer? Huh? Yes, sorry? Not audible.
iPhone.
Okay, you want to purchase iPhone. So which iPhone you will purchase? iPhone 13? 14?
Latest.
16? 17? 79? Sir is far ahead. Okay. So basically if you say, you can purchase the new version. Because the technology itself, it teaches us that how we should be acquaintized. Just an example. Have you done the online type of payments? Which mode you generally use for online payments? Huh? GPay. Yes, you do the GPay. Now my basic question is, suppose if your app is not updated and you are planning to do the payment, do you feel that payment is going to happen successful? Have you ever tried? Generally the system will say, you are using the old version. Please do update. The system itself is saying that get updated, get acclimatized, and if the system is saying on the same grounds, why won't we feel that there should be updation in the research field? And this is only feasible by the concept that is old versus new technology. Because in the old trends, what we have seen, we use the different types of the rodents, different types of the animals, they have been used since a long period of time. Now my question is, since last day, okay, you were studying, you understood the basic concepts related to what type of the research has to be carried out. Now just tell me, in the whole research, what do you understand the concept called as an enrichment, environmental enrichment? Yes, tell me. Is the environmental enrichment must during the any type of trials? Yes, because last day I think Dr. Vevare sir was there. He just mentioned the enrichment, how it plays a very important role. Okay, when we do certain sorts of trials in the animals. Okay, because if you pressurize them and whatever the results that is expected, definitely you are not going to get. Okay, on the same ground, we are moving to the new technology such as CRISPR, then humanized system, we are using the large animals, invertebrate models. So before that, have you ever heard about CRISPR? Anyone? Yes, do you know what is the CRISPR concept? Okay, some of the guys, they are knowing with the concept of CRISPR. Okay, I just will enlighten in short what exactly the thing is. Most of you are from masters and degree students. Yes, am I right? Just raise your hands. Yes. I'm just checking whether you are awake or slept. Okay, that is the part I'm just checking over here. So just tell me, you go for the thesis binding. Okay, once your project is completed, you go for the thesis binding. Suddenly the referee, okay, if he calls you and he says that your page number 2 to 4, line number 9, there is a correction. Okay, something page number 2 to 4 with the line number 9, there is a correction. And that correction, if it is not rectified, then you have to go for rebinding. What will happen? Okay, I will give another situation and the submission is within 1 hour. Is it feasible? Possible hai kya karne ko corrections? No, there is no chances. But suppose if I give a magic wand, okay, which movie was having that magic wand? Harry Potter. Sorry, Harry? I have watched the Punjabi version, I think so. Okay. So, that is a Harry Potter. I think that was something like Wingardium Leviosa, whatever that thing it may be. In the same way, this magic wand called as a CRISPR, it is having an ability to edit the DNA. Okay, to edit the DNA. That means there was a mistake in page number 2 to 4. Okay, line number 9. So within a second or within a tenure, within a short period of time, that correction is made. So in the same ground, this CRISPR, it is having an ability to correct your DNA and to remove the unwanted DNA, to rectify it, and that rectification is nothing but we call it as a CRISPR editing, or this is called knockout. It is removing the unwanted genes. And currently if you see, n number of diseases, the people they are suffering because of the wrong DNA or wrong gene which is functional in nature. Are you getting? So on the same way, if you see this CRISPR, okay, along with some enzymes, okay, GAG9, so I'm not going to go in detail, but certain enzymes are there, they're stitching, they're weaving the new DNA. And if it is brought into the picture, in the coming era, we can scan the embryo in the early stage, and whatever the child will be there, we can do the prediction also, that whether that particular candidate is eligible, I'm using the word eligible, for certain types of hereditary disorders. And that can be sorted out and that can be resolved. And nowadays we are using the animal models and different insect and worms on which these types of the technology is employed. Coming back. So why we are planning to shift to carry out certain types of the researches from the rodents, rabbits, or what are the other animals are used? By the way, can you tell me? For experimentation purpose, what type of animals are used? Yes? Snake? No? Okay, Guinea pig. Okay. Hamsters, monkey, dogs, zebrafish, okay, whatever. Now on the same ground, when there is a concern which is called as an ethical consideration, why can't we go for these types of insects, worms and fishes where we can successfully carry out higher version of the research? Because we are using these types of things, the advantages are the ethical considerations are quite easy. Simultaneously, there is a rapid life cycle. What is the life cycle of an housefly or fruit fly? It is around 40 to 50 days. So suppose if you want to do some genetical studies which may be carried from the particular generation, within the short period of time you can get the results. Instead of waiting for a year. Are you getting? Once upon a time, the great scientist George Mendel used to perform the experimentation on, yes, pea plants. At that time he didn't got that recognition, but his expertise, his ideas, they were so much innovative that nowadays also we greet him as a father of genetics. On the same ground, if we see the rapid life cycle or genetical trackability, so we believe that it is feasible but not replaceable for going from mammals or from rodents to insects, worms or fishes. Because currently the basic problem that our society which is facing, it is related to the diseases, toxicological issues and neurological and degenerative diseases. Now for example, suppose if you want to compare these two parts, nematode parallelly with the housefly, if we go for 3 generations, within the 3 months we get the results instead of waiting for 3 years on rat or mice. So that is called as a fast smart work. Similarly, when we talk about the fruit fly, that is a Drosophila. So in this case you will see 75% of the human disease causing homolog are present in this fruit fly. What is the miracle? The miracle, it means this is the evolutionary changes which has taken place and which is stored into this small insect. So if you talk, so we can go for the detailed studies which includes neurodegenerative diseases or even the cancer. For instance, suppose if a particular drug it is showing some untoward activities, because nowadays the basic problems which is seen is during the studies, some of the medicines they are showing some sort of cognitive disorders, and that can be studied easily in these types of insects. For example, fly VR. Do you know what is VR? VR system, virtual reality. आपको पता है virtual reality क्या होता है? Nowadays you go for 7D theaters, 11D theaters और पता नहीं क्या क्या theaters होते हैं. What you do generally, you cover the face with some sort of like, क्या होता है वो? Yeah? क्या होता है? It is some like helmet like system. वो हम लगाते हैं and accordingly we can visualize the things. Same things has been carried out, in which what we are doing, this insect, okay, it is mobilized on a system and we are running some visual things, and this insect it is reacting accordingly, and that it is going to help us to understand that what sort of the things, degenerative studies, cognitive abilities, all these are being studied by using this simple fruit fly. Another important part, we have microfluid arenas and automated behavior tracking system. I am using some vast AI-based like system, ideations in front of you. But what exactly the concept is? Suppose if you are brought up into a system, in that system, okay, उस system के अंदर हमने क्या change किया? We are changing the temperature, we are changing the pH, we are changing the locality, and we are seeing the impact. And that impact, if we get a particular result, definitely these results will be helpful to understand that what sort of the analytical part can be governed, brought up into the picture during the clinical trials. Okay, even the small activities like flippering activities, movement of their appendages, that also can be recorded, and this can bring a revolutionary change. Because last day we heard one PPT that nowadays for the milking of the animals we are using the musics, and the results are out of the imaginations. On the same ground, these small things has to be studied, because already the Paul Enrich, he mentioned the concept of magic bullet. Have you read the book of Magic Bullet? Okay, in that he mentioned every pharmacological moiety which we call as a xenobite. Okay, they have an ability to go inside the body with the ADME concept. Okay, but just remember, every molecule that enters inside the body, it is a foreign material. That's why the results or the activity or the PKPD, pharmacokinetic and dynamic parameters, they are totally changing. And that is the reason nowadays you can see the cases of ADR are very high. Because a particular molecule, if it is a boon to someone, so for someone it is like a curse. It can result into anaphylaxis or whatever the things are there. Similarly, this is my favorite part. It is one nematode which is called as C nor habitis elegans. Okay, it is not the title of some Tamil movie. Okay, so it is the name of one nematode, a worm. This worm, it is having 3 not 2 neurons, and these neurons, they are, and because of the short lifespan, we can go for aging studies. We can also have the neural circuit studies. What is the neural circuit study? For example, you are familiar about the synapse? Do you know synapse? So, synapse का एक simple example बताता हूँ, then you can understand in the better way. Suppose if you are made to sit with your so-called friend, okay, in some sort of like lectures and all, you enjoy it. I'm talking about the lectures. You fully enjoy because you are sitting with your so-called friend. But on opposite side, suppose if I change the sequence and if I made you to sit with someone जिसको आप पसंद ही नहीं करते, एक दूसरे को मत देखो. So in that case, just tell me, which is the feasible part? पहिला वाला या दूसरा वाला? हाँ? अपन जनरली म्हणतो ना एक नंबर. Okay. So on that grounds, if we change some sequencing, okay, by CRISPR, again I'm bringing that CRISPR editing part, and if I modulate the neuron, and if I check the toxicology and pharmacological studies of some new moiety, within some few months I can get a whole data, admit data. Okay, that is admit. What is admit? Absorption, distribution, metabolism, excretion and फिर से elimination. Okay, that is toxicological studies. So that can be studied by using this nematode. And what exactly is the advantage of this organism is, we have the complete neural map. We can play with it. We can do the things as per our wish. Simultaneously, we have a transparent body. We can see the activity inside, what exactly is happening. If we use some trackers, okay, some markers, so that also can be studied by using this type of nematodes. Parallelly, this organism is the luckiest organism. Why? Because it has traveled to the space. Because International Space Station, they have investigated the microgravity activities by using these types of worms. What exactly it means? Just simple example बताता हूँ. How many of you are like familiar about the concept called SpaceX missions, Elon Musk? Okay, most of you are familiar with that part. So what I'm trying to tell over here, these organisms, what exactly type of protein they can secrete, that study has been done at the space station. Because आप अगर इधर खड़े हो, you have some sort of G-force. G-force it means what? Gravitation. Okay. And when you go to the space, okay, when you are lifted from a particular point, your G-force it will become twice. Are you getting? And when you go exactly at the last point into the space, your G-force will be, कितना होगा G-force space में? 0. You are floating. Are you getting? You are floating. Now just imagine, when the astronauts they are going into the space for a particular period of time, okay, the cells which are showing the normal activity, they change. Because the force which was exerted on the muscles, the force which was exerted on each and every body part, it gets changes. And simultaneously the protein secretion or the basic metabolism, it also gets changed, which is called metabolomics. It is another study. So by using this nematode, they understood which type of the protein synthesis were restricted, what were the changes, and this is what a new modular study, because taking rodents and some sort of like humans into the space and doing the research, it is a costly affair. But rather within a short period of time, if you are using this, definitely we get an idea about muscular degenerations, neurological changes, altered aging and lifespan, reproduction development, gene expression, and the last important, nothing but the radiation effect. Because in the space, if we see direct, we don't have ozone. We are directly exposed to the what? The rays which are coming into the space, and that may damage your DNA also, which is not seen. Are you getting? So that also can be analyzed. And lucky part is that these organisms are so much adaptive. Whatever the rays were there, so they created certain types of proteins which are restricting, preventing it from the damage. Next, I think he is the role model to give all the details about the zebrafish. So it will be injustice to the audience that I am giving the details about the zebra, because I think in the afternoon he is going to conduct one session where he will give the brief idea why the zebrafish is used. But in short, I will give some glimpse. It is having a transparent embryo. It's a very beautiful type of a creature. Within the 3 to 4 days, you can see the egg, which is transparent. In that, you can see the blood vessels. You can see the heartbeat. Okay, and by default, suppose if you are injecting some drugs, beautifully you will see how the results are there. Are you getting? So that miracle we can see later on during the experimentation process. New tools which are nowadays used, we are using the light shield microscopy. That is a 3D imaging. It is an imaging technique where the light is inserted, it will create a 3D structure. That 3D structure definitely will help us to understand what exactly happens to a particular drug when it goes inside some biological system. Parallelly, we have automated embryo injectors which can inject the exact quantity of the drug at a particular point where there will be zero errors. This will minimize, the errors which can happen. Simultaneously, another part is that it is having a, I think sir, what exactly the count of eggs they are laid by zebrafish? It is in hundreds. It is in hundreds. Okay, so that is the part you can get the n number of the candidates for the further experimentations. Another, this is called planaria. I don't know how many of you heard about the planaria. I generally has searched the literature, old literature, and I came across one concept which is called as a Rakta Bij. You have heard about the one Rakshasa called as a Rakta Bij? When he used to drop a single blood, okay, on the floor, one new demon is to come. Okay, on the same part, this planaria, it is a living legend. If you cut into 99 pieces also, it will regenerate again into the new planaria. So just see, any neurological disorders if you are coming to see in the coming days, okay, it may be Alzheimer, dementia, Parkinson, so that can be studied easily by this planaria model. Are you getting what I am trying to say? So that means we can study this planaria because the whole body is having the regeneration ability, whole body. Okay, and here also we have some tools like laser microsurgery system which will remove a specific tissue, because we are trying to see whether if a particular tissues are removed, really it is going to regenerate. Surprisingly, it is regenerating. Parallelly, we can silence the gene by RNA electrophoration method. So to check whether by silencing some of the genes, really the regeneration ability is affected. Are you getting? So this is the another part. So coming back to the next mode, I'm going to wind up with the things because many other legendary peoples are sitting over there. Already we studied the old version and new trends, that how the rodents, what we used to do the study, it is automatically been replaced by the new technologies. Now I am going to talk about a further step, and that is nothing but the genetical engineering and data collection. Priorly, what used to happen, if you give the drug to any of the rodents, there will be a random mutagenesis and issues will be pertained into that particular organism where the desired results we used to get. Okay, whatever the expected results were there, we used to not to get. But by using the precision gene editing, what is the tool name? Gene editing? Yes, it is a CRISPR. This CRISPR can knock out the unwanted genes and they can knock in. That means we can bring inside the genes which are important. Okay, for example, certain genes, they are producing some sort of like insulin, some sort of like diseases are there where there is a glucose intolerance. Are you getting a simple missing enzyme which is responsible for a big disease? And CRISPR, if you see, you are fortunate that we have got the green signal from FDA to carry out the CRISPR studies in sickle cell anemia. Okay, but it is a very tricky process. Okay, it is a very tricky process. It is a very costly affair. But yeah, many researches are in process. Simultaneously, previously we used to terminate, we used to do the euthanasia, we used to do the manual sampling. But nowadays it has been replaced by the wireless telemetry. Okay, I am going to talk in very short what exactly the wireless telemetry in the upcoming slides. Now these are some research papers which are published and they are claiming that we are going ahead, a new mode, a new version, a new era where the results are fascinating. For example, this is one type of high resolution live imaging technique. In this technique we are using the primates, we are using the transgenic pigs, mini pigs. What exactly we are doing? We are peeping inside that particular organism, we are peeping inside that particular animal and seeing what the drug is doing. Previously we used to do the sampling, we used to wait for the hours, we used to see what are the results. But now merely by checking the organism we are getting the results. And how? By surgical implantation of cranial, that means you know stereotaxis. Okay, we remove the skull, we just do the things, blah blah blah. Okay, on the same way we are surgically removing some part, we are implanting some systems, and we are sending inside the radio labeled drug. And this radio labeled drug, what exactly it is doing? It is been showing the real time image, and parallelly what exactly we are getting? We are getting the results within the week without disturbing the animal. So that is the plus point. Simultaneously, so you can see the another updation. This is called high resolution positron emission, we call it as a PET scan. Okay, which is called micro PET scan, where we are using different types of animals. In this case you can see a radio labeled. Okay, we are using the radio labeled some sort of like ions. For example, we are using the C11, that is radio labeled carbon, with the drug analog, and that it will injected inside that particular animal. Once it goes inside the animals, what we are getting? We are getting the 3D reconstruction model, and in this 3D reconstruction model, we are getting all the data, including your allergies, including your side effects, adverse effect, toxicity. Okay, one question will be there. You can just note it down. मला फक्त नंतर कोणतं सांगा. Just tell me the difference between side effect, adverse effect, and toxic effect. Because if you are getting the answer for this, I believe that half of the pharmacology you have studied well. Okay, so this is the important part. So we can get all the informations, and the basic application here is we get the CNS drug biodistribution, which is a very important part. Because when we talk about the blood-brain barrier, this is the basic thing where the most of the drugs are rejected at a particular period of time. But if you are getting the idea, definitely it will be very much useful. Similarly, there are various other protocols. I am not going to go in detail, but just will tell that suppose if you are using fMRI system, in this system we are checking the results on the primates, on the sheep, by keeping them awake. Because if we do the anesthesia, the probability index is high that whatever the results which are coming by the drug interaction should not happen. And that is the reason these upcoming trends are coming, and you can see the research paper with the DOI already they are published. It means the research it is in process. Okay, it is ongoing, the results are out. Okay. Parallelly, we can have the various types of other sensors like in vivo electrochemical biosensors. In this, we are going to implant by stereotaxis. We are going to open the skull. We are going to implant some of the carbon fibers. The carbon fiber, the size is less than the small hair. That is implanted, and here we are going to go for the wireless telemetry. And this I am going to wind up with this part only. Wireless telemetry, it means we are inserting the sensors. These sensors will have some sort of receiver. That receiver, it is going to give the results wherever you are sitting. You may be sitting in the lab, you may be sitting in the garden, or you may be sitting elsewhere directly without disturbing the animal. Okay, we are getting the results. Second, just you can see some of the images how the experimentations nowadays are carried out, okay, by using this wireless telemetry system, because previous slide it was talking something related to brain. This is totally talking about the cardiovascular system and respiratory system. Are you getting? So this is the new era of the research. Simultaneously, I think Jarak sir is not available here, but he will give the brief illustration that how the electrodes play an important role for detection of the different types of the body parameters. So here we can do the implantation of 128 to 256 channels inside the brain, you can see over there, and that signals they can be received continuously. Okay, so that is the application part. Simultaneously, there are many models are there. Okay, I'm not going to go in detail. There are various models are available in the market where we can get all the details like glucose analysis. Even we can MRI, ECG parallelly to get the result in one stroke. Even we have one socio study which was carried out. Here we are going to do the implantation of some sort of like biogrids. These grids is going to check with the tag, that is RFID, that is radio frequency identification tag, the behavior, the social behavior of the particular animals. Because if the medicines are given, if it is treating the disease but social behavior is not appropriate, then how the things are going to work? Okay, so these are the upcoming trends which are coming into the market. As I told you, the large animals are nowadays being used with the CRISPR editing diseasing models, where we are checking each and every possibilities where we can treat the different types of the diseases. And the last part, now we are integrating our research with the AI. By using the AI tools, by generating the algorithms, we can predict the results with the proper accuracy and precision value. And once we get that accuracy and precision, then definitely we can opt with the traditional method for the final authorization if it is needed. So this was the in general information related to the modern techniques that can help a researcher, a young mind, to take a leap into the new era. Thank you.
Thank you so much, sir, for informative and enlightening session. Now I kindly request to participants to ask if any queries are there.
So my question is about, as you mentioned, that the shift from rats and mice towards the worms, insects. Specific about that particular insects, that 75% is homologic in the disease genes. But how can we justify the differences in the anatomical body systems or other systems?
Basically, when we talk about the different models, okay, there are various models available in the market. I'm telling about market in sense research area. So if you want to focus related to genetical studies, okay, so in this case we are employing some models. Even we are doing some anti-cancer model. For example, if you are doing some anti-cancer model, for example on the rat, by using some sort of like chemicals, you might be aware like DMH, dimethyl hydroxine, and that. So in this case, we are targeting a particular area. We are not targeting the whole system. And whenever the results are coming, then we are checking for the other parameters. So if we talk about the genetical type of a research where the less cost can be implemented, suppose if you have thousands of drugs molecules where we have to select for like 10 or some hundreds, for that case these types of models are very useful. This is used only for scrutinizing the molecules instead of like taking the n number of animals and getting the results. So for that scrutinization purpose, we are using these models.
Sir, you just tell about the fruit fly and nematodes. How handles the fruit fly and dose is how dose administration is calculated? According to the rat weight, dose is calculated. In this fruit fly, how dose calculated and how administered this dose?
Actually, the protocols vary from models to model. For example, if you are going for rhesus monkey, the dose can be by the oral route or by the other means. Suppose if you are going for nematodes and all, the media is there. So in that in situ media, whatever these worms are there or some insects are there, they are going to stay over there. And a particular period of time, they have some resting period and all. In that, whatever the drug content is there, that is been absorbed. And that parameters is being checked by radio labeling. As I mentioned, there is a radio labeling system, and that can be analyzed, and by default we get the results.
Yes.
Okay.
Okay.
Okay, thank you so much.
Now I request to volunteer Apeksha to start the puzzle session.
Thank you so much, sir, for this informative session, and we are really grateful to have you here. So, delegates, once again we'll start with the question answering session, but a twist ke saath. Now, I won't be telling you the options, you yourself have to give the answers, and I repeat, do not disclose the answer, please raise your hand first, okay? And the first question is, what is CRISPR-Cas9 primarily used for? Yes? Yes, that's correct. Please come forward. Please introduce yourself first.
Thank you. I am Sabashi. I am from Channabasweshwar Pharmacy College, Latur.
I request to speaker, please kindly hand over her gift.
Sir, what?
Actually, as we are running out of time, we can do one thing. So all the questions will be entertained by each and every jury who are sitting over there, and we are pleased to connect you. Okay. So in the afternoon we have ample amount of time during the lunch, so definitely we'll connect all the students. Okay. I'm not talking about me, but all the juries who are sitting over there or the speakers. Okay. Definitely will be.
You can ask the queries after sessions. Personally, you can meet to the speakers. Thank you.
So the next question is, which animal model has transparent embryos and is widely used in toxicology studies? Yes, ma'am.
Zebrafish.
Yes, that's correct. Please come forward. Please introduce yourself first and let us know your experience in this session.
Hello everyone, my name is Parveen Nada and I learned the zebrafish study from my guide Dr. Sandeep Patil, and I am working as assistant professor in Dr. J. J. Magdum Pharmacy College.
Please come on the dais.
Sandeep sir, please go ahead. Please take it.
Thank you so much, sir. Now I request Dr. S.S. Patil, sir, to kindly present the brief summarize the session. Please, sir.
So it's my pleasure to be here, and very first I would like to thank Dr. Janway sir appointing me as chair this session. In fact, this 2 days seminar is very fruitful one, and whatever I have attended the morning session and Abhinandan Patil session, really it's great and very informative sessions. And Abhinandan Patil is a very genius one. Is many prizes awarded person, and no doubt in what way he has been explained the future of this animal experimentation. So he has included all the things, animal models, genetic engineering, data collection, and various types of scanning and interpretation by using PET or MRI and so on. Definitely the coming days, it is the days of AI and machine learning, and definitely with these newer tools, the research will be very easier one, and definitely it will be a very precise one. And one thing he has been introduced, that is CRISPR. This is, I think, a very new term for all of you. And really with this kind of a research, we can go to the very specific results. So I congratulate Dr. Abhinandan Patil sir for his great lecture. And once again, I thanks all of you and wishing you all the best for remaining sessions. Thank you. Thank you so much.
Thank you, sir. I request you to kindly felicitate today's speaker, Dr. Abhinandan Patil, sir, with a bouquet and memento as a token of love and appreciation. Please, sir. And now I request Professor Jarad, sir, to kindly felicitate today's chairperson, Dr. S.S. Patil, sir. Please, sir. Thank you so much, sir. Now we'll move the next session, and it is my great pleasure to honor the welcome next speaker, day 2, session 3. It is my great pleasure and honor to welcome our next speaker, day 2, session 3, Dr. Balwant Chaure, sir. I request to Dr. Balwant Chaure, sir, to kindly take the dais. And this session will be chaired by Dr. Sandeep Kadsi, sir. Please, sir. I request Dr. Rameshwar Adar, sir, to kindly introduce today's eminent speaker, Dr. Balwant Chaure, sir. Please, sir, take your seats.
Hello. Good morning, all the respected dignitaries, colleagues, and my dear friends. Myself, Dr. Ramesh Vardar, Associate Professor in the Pharmaceutical Department, D.Y. Patil College of Pharmacy, Kolhapur. So it's my great pleasure today to introduce the eminent guest, Dr. Balwant sir. He is a senior academician in the field of pharmacology. So we are talking about education. Dr. Chaure sir is completed MBBS in the Government College of Medical Ambezawai and the MD in pharmacology from Mahatma Gandhi Institute of Medical Science, Sevagram, Varda. So we are talking about its profession. Sir completed 11-year teaching experience in the different institutes like Mahatma Gandhi Institute of Medical Science, Sevagram, GMC Dhule, GMC Kolhapur, and Dr. V.K. Patel Medical College, Ayanagar. So we are talking about the expertise. Sir having a vast experience in the clinical research, clinical trial monitoring and pharmacology. And currently sir working as a professor in the Prakash Medical Institute and Science, Ishwarpur. Sir having a number of several activities as well as courses like basic course in medical education, advanced course in medical education as well as basic course in biomedical research and many more. Sir as well hold academic positions as well as admission positions like academic coordinator as well as ADR monitoring center coordinators as well as secretary in the pharmacovigilance committee and GMC Nandurbar, as well as sir publishes 11 research paper in the national as well as international journals. So such eminent dynamic teacher with us. I warmly welcome sir. Please. Thank you sir.
Very good morning, one and all. So, I have been allotted the topics, that is animal handling, the routes of drug administration in animals, and the blood collection from the experimental animals. Since I know that animal handling has been covered yesterday afternoon, I think, hands-on training it was there, and then the routes of drug administration that we cover shortly in the same topic, that is collection of the body fluids. So after such a advanced and futuristic session, the previous session was very good, advanced one, futuristic one. So again we will come back to the traditional methods, how we collect the body fluids from the animals. So basics, I think that has been covered already. So what actually we do through certain animal studies, we first introduce that this is in the animals, so that to see actually how that animal behaves in that condition. Okay, and then accordingly we introduce certain drugs by different routes of drug administration. Accordingly, the same route that we are going to administer in the human beings, that may be the oral, that may be the inhalational route, that may be parenteral like IV, IM, intradermal, subcutaneous. Same way we administer such drugs in different animals. That is easy to administer drugs by all these routes in larger animals or the like monkeys, dogs, and cats. But that is quite difficult in smaller animals like the rodents. Much difficult in the smaller animals. Again, the zebra fish and the flies and nematodes and all those. In that case, we administer drug to the surroundings. That is, suppose for the smaller or the very smaller animals, we introduce drug by national route mostly, or we administer drug in the water they are swimming in, so that the drug will enter inside their body and the action will be seen. So here we start with the session, that is techniques for collection of body fluids in various animals. Why we need to collect the body fluids? To see the different parameters or aspects of the drug itself. So right from the beginning, we observe or we monitor the pharmacokinetic parameters of the drug, that is ADME, absorption, distribution, metabolism, and the excretion as such. Then different biochemical parameters like the serum investigations in terms of the glucose level, in terms of different enzyme levels. Okay. Then as well as the different toxicological studies, that what actually that drug will do when it enters inside the body, how it affects the liver, how it affects the kidney. That will be monitored by withdrawing various body fluids from the animal, and that will be analyzed, investigated by different tests, and the results will be accordingly monitored. Okay, so what are different body fluids? We know that we can commonly commonly withdraw from the animals. The most common being the blood. Okay, then less common fluids are the there may be then saliva may be there, urine may be there, fecal matter may be there, semen may be also there. Okay, peritoneal fluid may be there. Okay, so we'll see one by one. So again, what sample or what fluid should be withdrawn from that animal? That is again decided by what actually the parameter or what actually the topic of our study is, what we are studying. Okay, depending upon the animal as such again, and all this again coming to the fact that is again all the steps, the procedure that we are withdrawing the body fluid, that should be approved by the IAC, and that should be in your protocol, in your study protocol. So the various fluids that we know, that are the like from the blood, the semen, the saliva, the peritoneal fluid, the urine, the fecal matter, pancreatic juice, the biliary secretions, all those. We start with the most common, there is the blood. As such, whole blood can be withdrawn from the animal. It can be, if needed, it can be separated into plasma, serum accordingly, if you want to observe or monitor those things. To measure the blood concentration, as we already discussed, for the biochemical and hematological parameters. So whatever the procedures that we are going to perform to collect those body fluids, those should be less stressful to the animals, less pain should be there, less discomfort should be there, and that should be also handy to the person, the experimenter that he is going to perform. The required facility should be available at that center only. The technique depends on the species, the volume required, and the study design. Veins and arteries both can be used for the blood collection. Veins being the most common use. When we need larger volume, the arteries can be assessed in that animal accordingly. The blood collection method should be approved as I will discuss in the IAC. There should be less painful and stressful, adequate training. The person who is going to withdraw that blood or any body fluid, he should be skilled in that procedure, in that practice. Okay, he should be skilled. And then again, how much we can withdraw the blood, that we also will go through the one mathematical calculation for that calculation of the how much the blood volume we can withdraw. So for example, if any one of you have donated the blood, how much we donate the blood? It is around 350 ml. Okay. So why that comes so? How much blood volume we can donate? How that is calculated? So that is based on the weight around about 7 to 8% of the body weight. It is the total blood volume of the person. Suppose I am weighting, say my body weight is 70 kilogram. So my blood volume would be 7% of the 70. Okay, that is around 4.9. Okay, liters accordingly. Same for the other animals. Suppose for a rabbit, approximate body weight is 1 kilogram. The blood volume will be 7% of that is 70 to 80 ml. That is the total blood volume of that animal. And out of that total blood volume, 10% we can withdraw at a single setting. Okay, that is around 7 to 8 ml. And same for the other animals, depending upon the body weight. Body weight varies. These are the standard body weights that are mentioned here. However, the body weight of each animal in each group varies. Accordingly, we will calculate how much blood volume maximum we can withdraw. So this is a calculation, same as we have already discussed. Suppose for example, a rat 200 gram, 7% of the total blood volume would be the 14 ml. Out of that, 10%, that is 1.4 ml, can be withdrawn. If we need more than that, we need to replenish the volume. So that means we need to add, or we need to administer certain IV fluids, most probably the ringer lactates and certain colloidal preparations. One can administer so that it will replenish the volume which we have withdrawn from that animal. This is again same for the different animals, depending on the weight, the total blood volume, and the blood that we can withdraw. So what are the general principles here? That we have already discussed, the 10% of the total body volume. Out of that, 1% can be removed. In single withdrawal, 10% of the total blood volume can be removed. 1% of the total blood volume can be withdrawn in a 24-hour period. And we have to allow some recovery period in between. Suppose we remove 10% of the total blood volume, then we have to allow some recovery time in terms of mostly the 2 weeks, so that the animal will recover itself. And there are different methods of the blood calculations. On gross we call it as non-terminal methods and terminal methods. Non-terminal means we withdraw the blood and the animal survives. Okay, somehow we withdraw less amount of blood and the animal itself recovers the lost blood volume, or if needed, we can replenish with the external IV fluids like the ringer lactate, and the animal survival. That we call it as a animal doesn't die there, so that we call it as a non-terminal methods of blood collection. While terminal methods where the animal actually dies or we have to sacrifice that animal, that we call it as terminal methods. That we directly withdraw the blood from the liver, we withdraw the blood from the heart itself or from the vena cava. Okay, that means here actually animal will get died or the animal will be sacrificed. So that those are called as terminal methods of blood withdrawal. Here steps are important, that is what the animal should be handled carefully with a less suffering, less stress to the animal. Then most of the restrainers are used. You might be knowing the various restrainers for the rodents as well as for the rabbit. Then needle size, accordingly it is a common rule. Needle size should be smaller than the size of the vessel or vein itself. Always, it is the rule. It should not puncture the vein, so that's why it should be smaller in size. The site, most commonly the site from which we administered the drug, same are the sites for the blood collection or vice versa. Okay, larger veins are most commonly used, like the tail vein, the syphilis vein, okay, for the blood collection as well as the drug administration. Then for terminal methods like heart and the vena cava, the marginal veins of the rabbit, ear. And one more step here is the dilatation of the vein. So normally animal, the veins may not be dilated, so it may not be visible by naked eye. The needle cannot be inserted it with the confidence. So we have to dilate those veins. So by increasing the temperature, by keeping that area warm, by lightening it, by putting light over that, by certain lamps, by putting warm cloth over that, or by administration of certain chemicals like ozylene. Okay, that it can be certain anesthetic as well. It can be dilated, so that now vein will be dilated, it will be engorged, and we can put our cannula or needle in that, so that to withdraw the blood. So all the steps will be same for the each and every blood collection for the from different animals as well. Animal restraint will be the careful handling, anesthesia, certain blood collection from certain sites might need the anesthesia. Suppose we are collecting blood from the retroorbital plexus, so here we need the general anesthesia. Okay, from tarsal vein where we need the local anesthesia. Okay, but there are certain possibilities that that anesthetic agent may interfere with the results. Okay, because what if we are looking for the certain findings, that is how it happens in the stress of the animal. Suppose animal is in stress, what are different parameters that are seen, or the biochemical readings that are seen in the blood. Suppose we are putting that animal on anesthesia before withdrawing the blood, the animal will be sedated, so the blood readings will vary. Okay. Again, there can be interactions, drug interactions. This anesthetic agent may interact with the drug administered. Okay. So these are different types of crystals you might be aware of. The needle size, as a rule, it should be smaller than the size of the vein. Ideally, the 17 to 27 gauge needles are used. The larger the gauge size, being the needle smaller, the needle bore. Okay. This 27B will be the narrower one, while the 17 will be the broader one. As already discussed, the dilatation may be done by the anesthetic agents, by warming it, by putting certain chemicals like ozylene, fentanyl, which is the opioid one, which can be also administered in rabbits. These are the common sites from which the blood collection will be done. The last one being the terminal procedures. The first two are the non-terminal ones. Out of that, first initial two are the which does not require anesthesia, that we commonly do, that is dorsal pedal vein, syphilis vein, and blood collection that requires anesthesia, being the tail vein, tail sleeve, or from the orbital spines in respective animals, jugular vein. Temporary cannulation can also be done for both drug administration as well as for the blood collection. These are the same veins from which we can administer drug as well, if you want to administer intravenously. Tarsal vein and the marginal vein most commonly in the rabbit. What are the requirements for that procedure? The hands-on training will be in the afternoon session. We will see the theoretical aspects of that. First we need the animal, the handling gloves, towel, cottons, collection tubes. Okay, the collection tubes here might be with the anticoagulant if we need whole blood, or like heparin, okay, or if you want to separate it into the plasma and serum, accordingly we can done without administration, without addition of this anticoagulant and by centrifugation method. Anesthetic agent, wherever the anesthesia is needed, warming chamber, the needles, 95% alcohol, that is nothing but our spirit swab that we administer to make that area, and as an antiseptic to make that area sterilized. Okay. Syringes of different size, electrical trimmer with most of the animals where the veins, the area, that site is covered with the hairs, that should be removed first. These are the different sizes that are the sizes of the needle that are used for the different veins. These are different anesthetic agents, right from the isoflurane to the ketamine, the xylene, which can be used as anesthetic agents for short anesthesia, medium anesthesia, long anesthesia with different needles. The procedure, this is the common steps for each and every blood collection. Accordingly, remove your superficial skin and debris over the that vein. Wherever needed, local administration of cream or spray of the anesthetic agent can be used 30 to 60 minutes before. Locate and track the course of the vein. Dilate and fix the position. How we fix the vein? By pulling it downwards, okay, or the distally, so that it will not slip. Same as that we do in the human beings while putting an intravenous cannula. Pierce the skin with a needle cannula. Collect the blood in reference container wherever required. Remove the needle and cannula accordingly. Suppose there are in same animal, it is a follow-up study. We are doing intermittent blood collection and same time admission of drug. We use two different veins. Same vein cannot be should not be assessed. Suppose in rat tail, like there are two lateral veins. One cannot be one can be used for the drug administration, and another one can be used for the blood collection. So that the results will not it will not interfere with the results. Once the procedure is over, one has to put the pressure over that vein accordingly by the cotton swab, by the fingers or hands itself, so that the bleeding will stop. And it should be also monitored in follow-up after some certain hours. Again, that animal should be monitored if there is again a latent bleeding is there or not, because that will cause again sepsis and other problems. Caution: suppose we put that needle in the vein and we suddenly pull the plunger backwards, so the gush of the blood comes in the syringe, but at the same time, because of that negative pressure, the the walls of the vein may get collapsed. So that should be gradual one. It should not be sudden one, because the veins are small, very smaller in size. So the hands-on training will be done in afternoon session. Possibly yesterday you might have observed the drug administration. Okay. Same here, the dorsal pedal vein of the blood collection. The steps will be the same. From the sequence vein, the method of collection, no anesthesia is required. Repeated sampling is possible. That is the advantage over here. That is mostly done in the mice and the rats. That is without anesthesia. These are the steps, same that we mentioned, the common steps. Accordingly, the blood will be withdrawn. No more than 3 attempts should be done, and collecting more than 4 samples in a day is not advisable. So this is a lateral circulus vein from which the drug can be withdrawn. Either once the needle is inserted in the vein, directly we can aspirate the blood by using the smaller syringe, okay, 1 ml syringe, or we will just remove the needle and the blood will ooze out, and that oozing blood will be collected in the capillary tube or directly in our bulb or the tube container. This is the capillary tube. The tail vein method, that is most commonly done by 2 methods, that is directly accessing the vein or directly cutting the tip of the tail. Okay. So actually that is a permanent amputation to the rat. So that is rarely tried. Mice and the rats. But the rest of the steps are the same. So here the difference between the mouse and the rat tail cross section, where the dorsal vein is present in the rat tail. Ventrally, both the tails have the arteries, so we never assess ventrally because it may injure the artery. Laterally, both rat as well as mouse, there are lateral veins. Rest of the steps similar that we have commonly mentioned, that is removing hair, trimming the hair, applying the anesthetic agent, guiding, choosing the vein, fixing it, okay, dilating it with the xylene or the warm, keeping it warm, or local anesthetic if needed, that can be administered. This is the retainer in which the mice is kept and accordingly the vein is assessed. Same for the drug withdrawal, same for the blood collection as well as same for the drug administration. Here the temporary cannula can be put where we want to repeated blood collection or repeated drug administration. Next method, this is quite cruel one. Rarely it is used by cutting the tip of the tail. This is again one more method where from a quite larger amount of blood can be collected, but it is again more stressful to the animal as well. So we need to anesthetize the animal completely. That is, we need a general anesthesia over here by certain agents like ether or other general anesthetic agents. We can anesthetize the animal, and accordingly we put that capillary is just behind the eye globe or in the retroorbital plexus where from the blood can be collected. It is inserted into the medial canthus just by the rotating method, and once the blood comes, it can be collected into the pipette. There is the position of the different veins is given there. So that is the retroorbital circle just behind the eye where we put directly the capillary tube. The lower one, that is the facial vein, where from also we can directly collect the blood by directly pricking over that vein. Caution should be the avoid repeated blood collection over here, damage to the eye, minor direction change or minor misdirection of that putting that capillary behind the eye in the plexus can lead to the permanent damage to the eyes of the animal. The complications for all the procedures where blood is collected, the complications could be the venous rupture may be there, then blood from this jugular vein. Also from the femoral vein we can collect the blood. Now the blood sample collection in the guinea pig, that is same as that of the rodents. Other rats and mice, that is cephalous vein or tarsal vein, cannulation or the cardiac puncture being the terminal method of blood collection. Tarsal vein, we have started with this only. The marginal, that is most commonly done with in case of the rabbit, where the rabbit ear has a very well prominent veins over the margins of the ear, from where we can administer drug as well as we can collect the blood. Okay, by two methods, either directly by the inserting needle in that vein and collecting the blood, or by cutting that vein by using the scalpel blade. By both the any of these steps, we can collect the blood. Venous dilatation can be done by warming it or hose hiding. And it is the image for by the needle, the collection of the blood, and here by the scalpel vein, we directly cut that marginal vein by the scalpel blade. Cardiac puncture and the blood collection for the vena cava being the terminal process where animal actually dies or we need to sacrifice the animal. Here we directly put that needle in the heart, most probably in the ventricle, so that the heart will not collapse. Okay. So here the needle is in the ventricles. So here this is the picture showing the connection from the posterior vena cava. So to revise, there are different animals. For example, in the mice, the blood collection is run through the vein, that is facial vein. We have the videos, we will go through that at the last. Submandibular or facial, cephalous vein or retroorbital plexus, most commonly. The cardiac, the terminal method in rat, tail vein, cephalous vein, the facial vein, and again the cardiac and jugular vein. In guinea pig or hamster, the cephalous and the cardiac being the rabbit, the marginal ear being the most commonly used along with the cardiac, and chicken from the wing vein or the jugular vein. So adverse effects I had already discussed, right from the minor hematoma, venipuncture, thrombophlebitis to aseptic conditions. Not from there can be the infection transmission or infection, or the wound may get infected. Okay, and severe complications will go on. Okay, gangrene may be there and much more may be there. Okay, phlebitis, scarring, nerve damage may be there. For example, retroorbital plexus, there may be optic nerve damage. Minor bruising may be there. Okay. Excessive blood loss may lead to the hypovolemia in the animal, so that if more than 10% तो जो 1% of the blood volume जो है उसमें से अगर निकाला है तो in that case we have to replace the blood volume or the volume as such, body volume, by replacing with the IV fluids. Stress related physiological changes may be there. For tail vein, the advantage being the repeated sampling as possible in retroorbital plexus. That should be done by only 10%. In cephallus vein, it is the best for the serial sampling of the bloods. For cardiac puncture, it is a terminal method. For marginal vein, it is easy and most commonly used in the rabbit. Precautions: not more than 3 attempts should be made. Continuous sampling should be avoided. Collecting more than 4 samples in a day is not at all advisable. And it should be always all the steps and procedures should be followed under the CPCA, ACA guidelines. Now the P has been removed, I think. So one should justify how much volume should be withdrawn, if that is actually needed or not, and that is actually the role of IAC, where we present our protocol. During the protocol, the IAC members discussion goes on, and they will decide or they will comment on how much volume should be withdrawn. If unnecessarily larger volumes are mentioned in the protocol, that will be corrected accordingly. The method will be corrected. Anesthesia, wherever the anesthesia is needed, that should be administered to avoid the pain or stress in the animal. All the records of such procedure should be maintained. Certain endpoints should be decided. After certain removing certain volume of blood, it should be stopped. After suppose there is a stress or injury to the vein, the collection should be stopped. Certain endpoints should be defined beforehand. The 10% should follow all these steps. Next, body fluids में blood के बाद आता है, that is urine. So urine or fecal matter we can collect by using the metabolic cage, where the 24 contamination free collection will be done. This is a photograph of the metabolic cage where the fecal matter stays above and the urine is collected at the bottom. These are different parts, right from the feeder, the water, the feed itself, that is provided in the metabolic cage. That is normally, naturally one can collect the urine whenever the animal passes the urine. One can manipulate the urine passage by manual stimulation over the pubic area, by just putting pressure on the suprapubic area or by applying the jeter pressure over the abdomen. Urine can be passed. Okay. By admission of certain drugs, for example diethyl ether, if we put that swab containing diethyl ether in front of the nostrils of the animal, it will induce the urination. And the procedure is quite larger animals, one can catch it directly and one can directly collect the urine sample. CSF, it can be collected by the cisternal puncture or the lumbar puncture under anesthesia. In pancreatic or biliary fluid, there are different methods by which we collect the fluid, and one has to separate the pancreatic secretions from the biliary secretions. There are steps in that also, so that we will get that fluid only, pancreatic fluid if you want, or biliary fluid if you want. The peritoneal fluid, that is collected by the paracentesis, and same procedure as by which we administered intraperitoneal injections. The synovial fluid directly from the joints, larger joints, one can easily collect this synovial fluid. Saliva, where that can be collected in the capillary tube. Naturally, animal saliva weights, in that it can be collected, or we will make that animal so that the increased salivation will be there by certain stimulating agent like citric acid. Semen also it can be collected for various reproductive studies like sperm count, sperm mobility, and all those. Okay, by the electroejaculation procedure or by surgically, that is terminal procedure, by surgically with sacrificing the animal and by collecting the semen from the epidermis and the vas deferens. So this is where all the steps and procedures and different sites from which different body fluids will be collected. Next, we can go through certain videos in very short. We will have a hands-on training session in afternoon for the same processes. But for any procedure, you should have a basic knowledge first, so that you will observe correctly and you will assist correctly and you finally will perform correctly. So whatever the holding techniques, animal handlings, what are different grips are there, what are different procedures, how to transfer one animal from the cage to the surface and to the lab itself, that are I think already discussed. We purposefully put sometimes animal over the lid of the cage so that that animal, it will grasp by its forelimbs to the mesh that is present over the lid, so that it will stabilize itself. अगर हवा में रखा तो it will move here and there. Okay. We'll put that in the restrainer. See here, the restrainer. They have developed their own restrainer from a juice bottle. Okay, suppose the animal is larger size and restrainer is smaller size. One can develop. You can develop your own ideas during research, and that's how the new inventions comes out. Right from the basic research, basic inventions like the restrainer to the advanced ones. Have any one of you have handled rat or mice in animal house sometimes? Anyone? Okay. What is your experience? If you directly go and try to handle it or try to hold it, it will hesitate. Okay. But if as in your research, if you go on handling it again and again after certain days, it will acclimatize itself. That every time you put it outside the cage, do something and you put it again. That means he gets acclimatized to that. That is, he is not giving, that person is not going to kill that animal. Okay, at the beginning you will feel some problems while handling. So the area has been trimmed by actually hair blade. There are certain electrical trimmers as well. The first, but that is step approach. He is going by the step restrainer. The area has been shaved or trimmed. Then that area is again sterilized with the cotton spirit swab. The vein is dilated and easily vein can be seen, and then he is going to put that needle inside the vein. Not more than 3 attempts. If each attempt fails to prick that vein, no, not more than 3 attempts should be made. One can try after certain time. Okay. जब तक vein में needle जाता नहीं तब तक try करते रहना नहीं. Animal gets stressed so that the findings will change and the coordination or the what you call it as a help from that animal will not be there. Okay. So he just pricked the vein and now he will correct in the capillary tube. After correction, he is putting pressure over that vein which is bleeding, so that and he will confirm that bleeding or oozing has been stopped. Again, after certain hours, he will again visit that animal house, that cage of the animal, and will check for if any there is that oozing is there. So the rat is kept in the restrainer. Gradually collecting the blood from the dorsal vein of the tail. Here the animal is being anesthetized by the ether inhalation in a beaker or container. Here, from the pedial canthus, the capillary tube is inserted by a rotational start. It apparently it is a cruel one, but comparatively than the other veins, larger amount of blood can be collected. If you want much more larger, the final method will be the terminal methods from the cardiac puncture and the vena cava puncture. After few minutes, the animal will regain its consciousness and will start moving here and there. Okay, next. This is quite simpler one. The veins are comparatively larger one. We can directly observe the vein and we can directly put the needle over there. The area has been trimmed accordingly as if cotton swab or spirit or alcohol has been already administered. If needed, local anesthesia can be given. The procedure doesn't end here. We have to stop the bleeding. So we have to put the finger and swab over there. So by applying certain pressure, that will be stopped. Local anesthesia spray can be used. Cream can be also used. This problem is from the artery when larger volume of blood is required. You can directly collect it from the tail artery. Facial vein, that is a submandibular vein. It is also the procedure to collect the blood directly by pricking just the marking the vein where it can be and forward. And directly pricking over that vein by the needle. See, by a single shot, he directly pricks over that mandibular vein. So after blood collection, ideally these animals should be caged separately. They should be kept in isolated single animal in a single cage if the facility allows you, or at least crowding should be there, so that the whatever the blood oozing out, whatever the injury, so that will again will not get infected, and the animals may bite each other's injury as well. Okay. So here comes the ends of the session. So I am very thankful to the organizing team, organizing committee for giving me this opportunity. Thank you so much. If any questions,
Thank you so much, sir, for sharing valuable insights on the role of administration and animal handling and collection of. Now, it's time to delegates. If you have any queries, you can ask. Okay, you can ask later directly to the speaker. Now it's a puzzle session. I request to Saima and Maria to start the session.
Hello everyone, I am Saima Patwekar.
I am Maria Bhagwan.
We sincerely thank our respected sir for the informative session of drug administration and animal handling and environmental enrichment.
The lecture was clear, practical and highly informative. Thank you sir for your valuable time.
So get ready to participate, stay alert and enjoy this quick game.
तो चलिए स्टार्ट करते हैं क्वेश्चन आंसर्स की सीरीज से.
डोंट वरी, ये कोई एग्जाम नहीं, सिर्फ क्वेश्चन पूछे जाएंगे. So be ready with questions.
The question will be based on key point discussed during the session. First MCQ is, what is the maximum blood volume that can be collected from a laboratory animal in non-terminal procedure? I read the question. Uh, what is the maximum blood volume that can be collected from a laboratory animal in non-terminal procedure? May I request to come forward?
Thank you everyone. Myself Shweta Patil. I am as a working as assistant professor at Sri Sant Kupa College of Pharmacy, Bhogam. And this technique I learned from my mentor, Dr. Sandeep Patil.
Thank you. Please come on the dais for the your gift prize gift.
I request to Dr. Balwant Chaure sir to felicitate the winner.
So second question is, which blood collection method allows the maximum collection?
नवीन चालते कोणाचे
Draw button.
No, that is wrong answer. Wrong answer. That is absolutely correct answer. Please come forward. I request you to introduce yourself.
My name is Pratisha Shinde. I am from Sri Sant Kupa College of Pharmacy, Bhopal. Thank you.
Please come on the dais for the gift. Thank you so much, sir, and thank you all the delegates for the actively participation. Please have a seat, sir. I request Dr. Sandeep Kadsi, sir, to kindly present the session summary.
Once again, thank you for this opportunity to summarize the practical insights which were given by Dr. Balwant Chaure sir on techniques for collection of blood fluids in animal experimentation. To briefly summarize, sir has given us practical insights on how to collect the blood from various sites, the significance of collection of blood sample, the volume of blood to be collected, which has to be kept in focus and in mind, the terminal and non-terminal methods, handling the needle sizes like 17G to 27G, and how to remember like which gauge size has to be used for which type of blood retrieval, the retroorbital plexus where we can collect large volume of bloods, and the pre-requirements for collection of blood samples. Apart from the blood sample, he has also focused on collection of samples like urine and how to stimulate so that the urine can be collected, the collection of CSF fluid, that is cerebrospinal fluid from the lumbar plexus, the synovial fluid from the joints, and saliva by inducing with citric acid. And he has also given what are the adverse effects and what are the distress conditions the animal will be facing when we are absolutely withdrawing the blood, like hematoma, phlebitis. And he has also given us a wide exposure like what is the significance of records to be maintained during this process of blood retrieval. So it was more of like a not vertical, virtual practical session, and this is like a preparative phase for the afternoon session, which will be a live demonstration. Thank you, sir, for such wonderful practical demonstration. I mean virtual. Thank you. We are honored to have you here.
Now I request to Chairperson Dr. Sandeep Kadsi sir to felicitate today's speaker Dr. Balwant Chaure sir with a memento and bouquet as a token of love. I request to Ms. Pooja Deshmane ma'am to kindly felicitate Dr. Sandeep Kadsi sir. Please ma'am. Thank you so much sir.
We move on to session 4. It is my great pleasure and honor to welcome our next speaker, Dr. Ravindra Jarak, as a speaker of this session. Please come up, sir, and take to the session. This session will be chaired by Ms. Sonali Divete ma'am. May I request Dr. Poonam Chogle ma'am to kindly introduce the speaker.
Respected dignitaries on the dais of the dais, esteemed resource persons, faculty members, delegates and my dear students, a very good afternoon to one and all. It is my proud privilege to introduce today's esteemed guest speaker, a senior academician and experienced researcher and a dedicated mentor who has made significant contributions in pharmaceutical education and research. We are honored to welcome Dr. Professor Ravindra Jagannath Jarak sir, a accomplished academician with over 25 years of experience in teaching and research in the field of pharmacy. And he is currently working as a professor at Bharati Vidyapeeth College of Pharmacy, Kolhapur, at Department of Pharmacology. Sir has completed M.Pharm from prestigious Government College of Pharmacy, Karad, affiliated to Shivaji University, Kolhapur, and also gained 2 years of valuable industry experience, giving him a strong blend of academics as well as practical exposure. Dr. Jarak sir has successfully undertaken 23 research projects and has received research grants and consultancy support exceeding 6 lakh from reputed bodies such as like Rajiv Gandhi Science and Technology Commission, Government of Maharashtra and Shivaji University, Kolhapur. His research contribution include 33 publications in national and international journals and 69 research presentations at various scientific forums. In recognition of his research excellence, Sir has received a Best Poster Award at an international conference held at NIN Hyderabad, along with Best Publication Award at BVVS Bagalkot. He has actively participated in 79 workshops, conferences and seminars and has recognized 27 faculty development program, workshops and social activities. His strong commitment to academic leadership and capacity building, Dr. Jarak sir has a life member of professional bodies such as the Indian Pharmacological Society, ISTE and APTI, and he is a registered pharmacist of MSPC. He also has his area of research and interest including preclinical and toxicological evaluation of drugs, in vivo and in vitro screening, wound healing studies, formulation development and screening of novel anti-cancer formulations. by making this expertise highly relevant to today's workshop on experimental animal handling on topic demonstration and application of physiograph by using Biopack M35 system. So, with this great respect and honor, I request Dr. Professor Ravindra Jarak sir to kindly grace the podium and enlighten us with this exposition. Please, sir.
Hello. Hello. Is it audible to you, everybody? Okay. Thank you very much for the Patil group and nice introduction by Poonam madam. Looking at your faces, I realize my presentation time within 1 hour continuously from morning session you are listening continuously 3 sessions. So I will consider that till your lunch break we will finish both the sessions. Okay, so be attentive. Rather than the lecture, today I am going to give the demonstration, live demonstration by using physiograph Biopack MP35 system. So before that, continuously in your experimental pharmacology laboratory, in the previous years we used to one instrument. I think everybody know that Sherrington revolving drum machine. Okay, nowadays it is just for a model only, nobody is using. But again, as a researcher, earlier to the physiograph, Sherrington machine is the only machine available to record all type of the responses. Okay, so physiograph word itself indicates meaning. Physio means physiological responses and graph in the form of graph it is represented. Worldwide, 2 companies are there, they are manufacturing this physiograph of good quality. One is Power Lab, which is US based company, and one is Biopack, which is UK based company. Cost of this instrument is around 10 lakhs rupees and it can record all type of the changes, it can record all type of the physiological changes inside the body. First 10 minutes, I will give the introduction of this instrumentation, types and application, and remaining 5 to 10 minutes with your one volunteer, I will demonstrate you, live demonstrate how to record one electrocardiogram. As far as I think one good example, what is reflex action, everybody knows that. Whatever changes inside the body, sensory nerve will pick up that impulses. Brain is there, brain is going to interpret that particular response. And motor nerve, they are going to give the effect to the terminal organs. Same mechanism is there in the physiograph. This is the basic machine that is called as a data acquisition system, BSL. By using this particular Biopack, it is available in the 2 versions, BSL Lesson and BSL Pro. BSL Lesson for the students for the demonstration purpose and BSL Pro for the practical purpose. So just like a brain, this is a data acquisition system. Whatever responses are there, all responses will be recorded in this machine. Secondly, this is called as a transducers. This is the one example. This is ECG transducer. What is the role of this transducer? whatever changes are there inside the body, these changes will be recorded either in the form of temperature, airflow, pressure, contraction, movements, beats, pulse, any type of the changes are there, that particular changes will be received by this particular part that is called as a transducer. This transducer is connected to the machine. Whatever changes are there, that changes will be picked up by transducer and that given to the basic system, data acquisition system, that is the BSL. Whatever changes are there, that will be recorded, implemented, and it will be converted in the form of amplitude. And this computer in which software is installed. This is hardware, this is software. In this computer, software is installed, and with the help of software, whatever data which is taken, that will be converted in the form of graph. And whatever is recorded, whatever you can display on the monitor, that you can save or you can take the printout of that. So that is the basic, means transducer will receive the impulses, data acquisition system which will collect and interpret the response, and software will be displayed in the form of computer, and you can record, and in the software itself we have n number of versions. With that you can analyze that particular results and calculate in the form of statistical significance. That is the basic. Now, as far as this data acquisition system is there, from the front side, channels are there. See, what is the advantage of that? This physiograph, initially it is available with a single channel. At a time you can record the activity, one activity only. So as a preclinical research, if any new moiety, new drug which is administered inside the body, that has to be given effect on multiple system. Even the drug is cardiovascular, it is going to affect on the other organs also. So while performing the clinical trials or preclinical trials, multiple times you have to use that. I think as far as the errors are concerned, physical error, chemical error and biological error. Okay, so for example, I have recorded ECG now, and after 10 minutes, again I am recording the ECG, you will get the different pattern. Same with the other pattern. So, due to the biological variation, to avoid the biological variation, for example, I am performing the study the effect of blood pressure. So, I am going to use 6 animals. Okay, so while recording the blood pressure, at a time, if single channel is there, I am going to record the blood pressure of one animal. 3-4 hours. Then second animal. Then third animal. 6 in the test, 6 in the standard, 6 in the control. So 3-4 days are required. What is the advantage of multiple channel? So earlier with the help of Sherrington drum, only one activity at a time. With the single channel, only one activity at a time. But what is the advantage of multiple channel physiograph? At a time you can record multiple activities. Whatever new drug you are going to study, administer a single dose and attach the transducer, multiple transducer to that particular animal or human. So, for example, this transducer having 4 channels. As far as the BioPack MP35 systems are concerned, 2 channel, 4 channel, 6, 8, 16, 24, 64 channel physiographs are also available. So what is the advantage of that? For example, the dose is given. So one transducer attached to the channel number 1, which will record ECG. Second transducer, which will record EOG. Third transducer, it will record EMG. Fourth transducer, it will record BP, blood pressure. So one animal at a time administered single dose, what is the effect of that drug on multiple organ? Myogram, EMG for muscle activity, EOG, electrooculogram for eye movements activity. Then EEG, electroencephalogram on the surface of your head. Attach the lead, it will record multiple graphs of electroencephalogram. To the chest, fix the leads, you can record electrocardiogram. Then EGG, electrogastrogram. Okay, then blood pressure. So with the one dose, you can record with the multiple activity at a time. You can save the time also, cost also, and you will avoid biological errors also. This is the biggest advantage of this transducer, this multiple channel physiograph. Today I am giving to demonstrate you regarding the ECG, but what physiograph available with us, with that we can record the ECG with the ECG transducer. Electromyogram. Okay, for example, in case of the myogram, transducers are like just like the needles. For example, these two hands, as per anatomy is concerned, same it is. Okay, but comparatively, my right hand having more strength than the left. So how we will check? Muscle is there, but muscle strength is there or not. For that particular purpose, you can insert the needles as a transducers in the muscle. You can record the electromyogram. Electrooculogram means graph related to the eye. Electroencephalogram. In case of the encephalogram on the surface, everybody has seen, to the head on the different surface the leads are fixed. Okay, and gamma wave, theta wave, delta wave, these waves are in the form of amplitude they are recorded. So by recording the electroencephalogram, you can check which area of the brain is affected. By recording ECG, I think everybody has seen the ECG. Okay, PQ, RST, theoretically, everybody knows. But it is very, very difficult to interpret that particular ECG. Okay, whatever positive waves are there, what are negative waves, what is the waveform, activity generated, that has to be properly interpreted and it will give the activity related or functions related to the heart. Second transducer available with us, that is the blood pressure. I think everybody know what is the definition of blood pressure. Pressure exerted by blood on the inner side of the blood vessel. How we are recording the blood pressure? Externally we are applying the cup and applying the pressure. So is it not objectionable? How means externally we are applying pressure and from back pressure, cup method we are recording. How, as far as the research is concerned, how you should record the BP? At actual, this transducer has to be inserted into the blood vessel. In the carotid artery, you have to insert the transducer. And then live you can record while normal blood is flowing to the artery, how much pressure is generated on the walls of the artery. Invasive method, non-invasive method, everybody knows, but as far as the preclinical research, clinical research is concerned, it is recommended to go for invasive method, that is called as a direct method to record the blood pressure. Second important, one more transducer is concerned, that is the body temperature. That transducer inserted into the body cavity, rectal, vaginal, you can record the body temperature. Next transducer is, force transducer is available with us. I think in the third year pharmacology, final year pharmacology, effect of drug on muscle contraction, relaxation, with the help of student or organ bath you are recording. Okay, and in the form of how much millimeter muscle is contracted, you are recording that. So with the help of force transducer here, in the form of gram, how much weight is required to contract or relax that particular muscle. So lively we can record with the help of piezogram. We have nerve conduction transducer also. Means for example, nerve cell is there, but all the nerve cell passing the impulse from one place to another or not. So by attaching with the help of nerve conduction transducer, you can check whether the nerve conduction capacity is good or not. This particular instrument, both way, clinically also preclinically also used. Preclinical means in case of the animals and clinical means in case of the human beings. Okay. So that is about the introduction and cost at a time means basic system will remain same, but this transducer will vary because data acquisition system will remain same, software hardware will remain same. Only what type of the activity you are recording, accordingly the transducer will vary. Okay. So we'll have the session of 10 minutes to record the ECG. Willingly, if any volunteer, please come on the stage. Just like a computer, you have to plug this to the channel number 1. I have fixed this transducer to the channel number 1. Please. I think everybody knows ECG, electrocardiogram, P wave, QRS wave, T wave. QRS complex is very important. Point P is positive, Q is negative, R is positive, S is negative, T is positive. To record the proper ECG, the patient should be resting position, lying down position on the bed. And whatever leads are there, that leads has to be fixed on the chest. So this is the open forum. That's why other method is there, because you know that why these waves are generated. P wave is generated due to the P wave is generated due to what? Contraction of atrium. Both atriums are contracted at a time, that's why P wave is generated. QRS wave is generated due to the contraction of ventricles, QRS wave. And T wave is generated due to ventricular relaxation. What about atrial relaxation? See, movements are 4. Atrial contraction, atrial relaxation. Ventricular contraction, ventricular relaxation. but the waves are 3. So which wave is absent? Atrial relaxation wave is absent. Why this wave is absent? Because that wave is superimposed by QRS. The time at which atriums are relaxed, that time ventriculars are contracted, and the intensity is more comparatively the relaxation. That's why atrial relaxation wave is superimposed by ventricular contraction. That's why atrial relaxation wave is not visible. Now here the whatever leads we have to fix, because there are a n number of positions. Minimum if you want to record, because in the standing position, sitting position, lying down position, leads are fixed on the different parts of the body, probably, preferably on the chest, and secondly on the wrist and limbs. Okay, so due to the contraction and relaxation, I think everybody knows that how the pulse is generated and you feel the deflection. So there are the certain points, around 12 points, where you can fix these particular leads. So that particular changes will be recorded by this particular lead, and that will be formed in the form of deflection, and it will be visible on the monitor. Why this is very important? If anything happens wrong to the P wave, cardiac surgeon or cardiologist will focus on the atrium part. If anything happens wrong to the QRS wave, cardiologist will focus on the ventricles. Right leg. This. A red lead, which is a positive lead attached to the left hand. Metal side has to be the bottom. White lead, which is a negative lead, which is attached to the right arm. And black lead, which is ground or earth, which is attached to the right leg. Be relaxed. In this software, first you select the channel. Already I told you, multiple activity at a time you can perform. Whatever a transducer which is connected to the object, which is inserted or attached to the data acquisition system to the channel number 1. So I have selected channel number 1. Once you click the channel number 1, this number of transducer list is available. So out of that, I will select electrocardiogram, ECG. Then set up data acquisition length. I think everybody know that with the one ECG you cannot interpret the results. Okay, once you fix the leads continuously for 5 minutes, 10 minutes, the recording has to be continuous. So you have to fix the time. So I will set here 5 minutes. Yeah.
Second channel. Channel.
tissue. Once you will click the start button, it will have the 4 sections of the graph. First section will be continuous ECG recording. Second section, P wave. What is the amplitude of P wave? Third section will be the RR interval. Every ECG having 0.8 seconds. So automatically it will record R to R distance, RR interval, and last graph will be the heartbeats. So whatever continuous recording is there, if anything happens wrong, we have the pointer here. You can copy that particular images, you can give the specific point, you can give the help of marker, you can object the specific point, and continuous recording, you can save that record. And in this also we have the journal where you can paste the data. This particular data in the form of values, in the form of graph. You can save also and you can take the printout also. Okay, thank you.
I request our volunteers to take the questions puzzle session now.
Thank you, sir, for the clear demonstration of the physiograph and its application in experimental animal handling. Thank you, sir, for your valuable time and guidance.
So first MCQ is how many channels are there Biomech MP35 system physiograph?
Correct. Answer is correct.
I request you to come forward and introduce yourself.
त्या डेलिगेटला प्राइज मिळालेला आहे. नेक्स्ट डेलिगेटला चान्स. ओके, कम फॉरवर्ड.
My name is Ruxia Farooq Sheikh. I am from Sri Samdukpa College of Pharmacy, Ghoga.
Second MCQ is which transducer is used to record muscle contraction? I repeat the question. Which transducer is used to record muscle contraction?
ऑप्शन द्या की.
Option A, electromyogram. Option B, electrooculogram. Option C, force. Option D, electrocephalogram. That was wrong answer.
Last.
Electromyogram is for strength of muscle. Question is contraction and relaxation of muscle. It is force transducer.
That was wrong answer. I repeat the question. Which transducer is used to record muscle contraction? Option A, electromyogram. Option B, electrooculogram. Option C, force. Option D, electroencephalogram. That was right answer.
The answer is right, but already Professor Dr. Jarak has declared the answer on the stage. So we'll cancel this question, okay? You be prepared for the next session now.
I request Ms. Sonali Lieutenant to kindly brief the summary of this session. Please, ma'am.
Very good afternoon, everyone. This is the moment of honor as well as gratitude for me. As the first footsteps of pharmacology I have learned from Dr. Jarak. So, beginning with the summary, in this session Dr. Jarak provided a clear and practical understanding of the physiograph and its application using Biopack M35 system. He explained how physiographic recording plays a crucial role in experimental pharmacology. The live demonstration helped participants understand not only the working principle of the Biopack M35 system, but also its importance in generating accurate, reproducible and interpretable experimental data, which will be again shown by the sir after correction of the system. Dr. Jaran has highlighted how such advanced instrumentation bridges the gap between theoretical and real-time physiological responses, making learning more application-oriented and the research-driven. On behalf of organizing committee and all the participants, I sincerely thank Dr. Ravindra Jarak sir for sharing his knowledge, his expertise, his passion for teaching with us today.
Graph started. Let it be. See, continuous movement, I think everybody has seen, directly, indirectly, to visit to hospital specifically in the patient is in the ICU. So for demonstration purpose we have attached this lead to the arm and leg. But if you want correct, see this now this is okay, is normal. But still if you want to clear ECG, that lead has to be fixed on the chest and patient should be lying down position. Then you will get the perfect ECG. Okay, and just like a software, everybody knows that this particular graph in the form of different forms of the graphs you can save in the form of figures, in the form of tables, in the form of pictures you can convert, in the grid form, in the journal form, you can copy and paste the record. Say, this particular data you can directly feed into the software and directly the software will give whether the results are significant or not. This is for the human, same way this lead you can use for the animals also. Thank you.
Thank you, sir, for an excellent session for demonstrating an application of physiograph by physically. May I now request Ms. Sonali Divatemem to felicitate the speaker with memento and bouquet as a token of
Professor C.R. Patil, is it audible? He is my batchmate. We have completed our graduation in Government College of Pharmacy, Karad from 1992 to 1996. I am honored to say he is my roommate also for 4 years. And now he is excellent. Okay, in the introduction, Sonali will give the introduction. He is a great teacher. Recently, he got the award from Prime Minister and President Government of India, Best Teacher Award of India. And he is the first pharmacy teacher got this type of the award. So our party is still pending. Party, sir? Yes. Thank you.
You all can see the passion of Jarak sir. We used to do pharmacology practicals for 9 hours at least. Sir did not allow us to leave the lab unless and until we perform. Each and every person in the lab performs the practical. Dr. Sandeep is the witness for the same.
Majority are there.
Yes, he is immediate senior to me. So 2 mentors are here today. Thank you both of you for being here, sir.
Thank you. Thank you.
Sir, please.
Sir, C.R. Patil, sir, can you just unmute for a second your mic?
Okay.
I request now to Mrs. Snehal Shilawat ma'am to kindly felicitate.
24/7 our lab is open. This physiograph is available to all budding pharmacists, researchers. Anytime you can contact for preclinical study. We have 6 to 7 different transducers. ECG, EMG, EOG, EEG, then blood pressure, force transducer, then cardiac output also. Directly place the lead on the chest directly you will get the cardiac output.
Thank you, sir.
I request now Sneha Shiravan to kindly recite Ms. Sonali Mittal ma'am.
Thank you so much, ma'am. Thank you. Now we'll move the next session, day 2, session 5. We are pleased to welcome Dr. C.R. Patil sir as a speaker for this session. Due to his busy schedule, sir will not able to join us physically, but this session will be conducted by Zoom meeting. I request to uh, this session will be chaired by Dr. Abhinandan Patil sir. I request to Dr. Sheetal Jadhav madam to kindly introduce today's speaker. Please, ma'am.
Good afternoon to one and all. Some people follow the trends, some create them, and a rare few redefine the entire fields. Our next speaker belongs to that rare and remarkable category, a distinguished researcher whose work has significantly advanced in the pharmaceutical science. Good afternoon to respected dignitaries, researchers, academicians and participants. It's been my great privilege to introduce our esteemed speaker, Dr. Chandrakant R. Patil sir, an eminent academician and visionary pharmacologist. Dr. C.R. Patil sir is currently serving as a professor and head of department at R.C. Patel Institute of Pharmaceutical Education and Research, Shirpur, where he continues to lead the impactful academic and research initiative. He holds a PhD in pharmacology from Shivaji University, Kolhapur, Masters in pharmacology from Gujarat University, Ahmedabad, and bachelor's degree from Shivaji University, Kolhapur. Dr. C.R. Patil sir is also proud recipient of several prestigious and recognition awards. Already Jarak sir also has mentioned that the National Teachers Award in the year of 2023 by Ministry of Education of India, New Delhi, Innovator Teacher Award by MHRD Government of India, Young Scientist Award by CCRH Ayush, and at the 6th World Congress on Alternatives to Animal Use, Rome, Italy in Life Sciences. Best Poster Award at the World Congress on the Alternatives to Animal Use in the Life Science. His scholarly work has earned almost 6,000 citations with an H index of 39 and I-10 index of 80. Demonstrating the strong global impact, his research contributions have not only advanced in the pharmaceutical science but also inspired young researchers like us across the nation. With over 25 years of huge experience in experimental and molecular pharmacology, his pioneering work in inflammation, fibrosis, and translational pharmacology has significantly advanced the scientific understanding. A major highlight of his academic and the research journey is mobilization of research funding exceeding 693 lakhs by a single person, supported by the recognized agency like AICT, DST, SARB, ICMR, Ayush and VCRMS. Notably, his landmark ICMR funded project worth of 4.5 crore on targeted therapy for NSCLC reflect the national importance of his research. Today, we are honored to have him for sharing his insight the stage and deliver his keynote address. And I am quite fortunate to the organizing team to introduce such an eminent personality who has been my government career senior all the time and for guiding me all the time. Thank you very much.
Hello, can you hear me? Please confirm.
Yes, sir.
Okay. Thanks a lot. I think there is some echo, but let's try to go ahead with that. No problem. I am sharing my presentation here. Hope this presentation is visible to all. At the outset, I am thankful to Dr. Janame sir, Jadhav madam, Abhinandan Patil sir for providing me this opportunity. And it is really a pleasure that I am talking after my roommate and my friend, Dr. Jalal sir. I am also pleased to know that Sandeep Patil sir was also presented before me. So you can consider us three as friends who are enjoying the same stage today. And without much ado, I would like to start with my topic. Thank you all for giving me this opportunity. The topic that I have chosen for this particular workshop is effect of environmental settings on the behavioral outcomes in experimental animals. Particularly what happens is when we are performing any pharmacological experiment, it is a great challenge because we are dealing with the most complex kind of system. That complex system is going to has hundreds of variables which are going to affect the outcome of our experiment. And that is the reason why all pharmacologists have to be very, very sincere, very, very sensitive while setting up pharmacological experiment. Many times what happens is because of lack of knowledge, because of lack of exposure that is needed, or because of some misunderstanding about the way we are performing experiments, we keep on carrying experiments in the conventional manner and we do not look at the intricacies which are there in the actual protocol. And that is where we may come up with slightly compromised kind of results. To avoid that, there is necessity of sensitization of the future generation, all but being pharmacologists and the people who are working in the field of pharmacology. Sit together and we should look at some variables which are very seriously affecting the outcomes without our understanding, without our knowledge. And that is the reason I would like to share with you few examples where environment, handling, the kind of bedding material that we are using, the lighting setup, how these things in the animal house or in the experimental room are affecting the outcome of our experiments. And such outcomes, just now we have seen a demonstration of ECG recording in human beings or in animals. There we are having a particular set of physiological parameters which we are trying to record. So when we are dealing with such kind of experimentation, like say blood pressure recording, ECG recording, there we are having comparatively more reproducibility in the results. But as soon as you shift to behavioral science, there the problems start coming. Why there are problems? Because behavior or psychiatric disorders themselves are quite complex. So in this particular discussion of 40 minutes, we will be trying to look at what are the problems with psychiatric disorder modeling. Then we will be looking at a very important aspect which remains neglected in the field of pharmacology, that is validation of experimental model. We rarely talk about this in academic. Then we will be taking anxiety as a model and a simple test of elevated plusness that is used for checking anxiety in animals, which we routinely perform. And using that particular example, I will try to explore or try to discuss about the factors which are affecting animals' behavior in this simple test. And then we'll try to see what inference we can get out of such kind of discussions. Discussions regarding how to perform a behavioral test, what kind of factors are going to affect our outcomes, and how we have to control as many variables as possible so that our results are more reliable, more reproducible. So this is the agenda of next 45 minutes or 40 minutes talk. Let's start with this particular psychiatric disorder. When we say psychiatric disorder, this is something very unique to human race. Okay? No one has seen a rat committing suicide. No one has seen a rat sitting aside and thinking worthless, or there is no feeling of worthlessness kind of things in the animals. This is something which is unique to human beings. Okay? And that is the reason why it is very difficult to create animal model, behavioral model for testing the drugs which are acting on psychiatric disorders. Whatever animal models we are using, those models or those animals, they are not having very well developed brain system or very well developed social behavior. They are having their own set of, we can say, biological activities which we can correlate with human social interactions, human anxiety, human depression. But then there is a sea of difference between what animals are and what we human beings are. And this is where modeling a psychiatric disease, as simple as anxiety, becomes a great challenge as far as behavioral pharmacology is concerned. So I think I have highlighted why it is difficult to look at behavior of the animals and correlate it with some disease condition in the human beings and then use it as a model testing drug activity. When we are dealing with psychological disorders, behavioral testing, we have to be very, very conscious about what we are doing and then whether we are maintaining all the conditions properly or not. Okay. Then let's come to the next important aspect of this particular behavioral testing. Validation in behavioral testing is still a complex process. If suppose we are talking about recording of blood pressure, then in rats we get 80-120 mmHg blood pressure. ECG we take, there in rats the heartbeat will be around 300 beats per second. In humans it is around 72 beats per second. But still the kind of responses which we are getting, EQRST curve which we are getting, they will be similar in animals and human beings. So we can very well correlate our recorded ECG of a rat or a rabbit or a guinea pig to human ECG or even dog's ECG to human ECG. But when it comes to behavior, we start facing challenges. There, the validation of experimental model becomes still more problematic. So before going to the details of this validation, we'll have to look at the meaning of term validation. This term validation we use for manufacturing related aspects or quality control related aspects, where we're talking about a process. Validation itself is a process which is ensuring that the outcome of that process will be uniform, it will be reproducible. You will be able to predict that kind of outcome which is coming out of that experiment. If you are having a set of protocol to be assured that the outcome of experiment is going to be a fixed outcome, reproducible outcome, then we can call it that particular process as validation process roughly. Okay. Talking about validation of experimental models, that to animal models, we have to look at the way in which we can validate these animal models. How we can become very sure that our experimental model is going to tell us exactly what we are looking at in that particular experimental model. Okay. So when it comes to pharmacological animal model and its validation, we are having different terminologies. Like first is face validity. Okay. Face validity means what? When we say that our model is having face validity, animal model is having face validity, it means that the animal model is showing similar symptoms which will be seen in a clinical condition in human beings. Means what? If suppose we are saying that a person is feeling anxiety, and we are saying that elevated plus means is a model of anxiety, then whether this elevated plus means is a valid model in terms of face validity? No. If this is the question, then we will have to check at the animal's behavior, how the animal is behaving on that elevated plus means. If its behavior correlates with clinical sign of anxiety, then we can say that our model is having face validity. I hope this is not creating too much confusion. When we are saying that our model is having face validity, it means that the symptom that animal is showing in that particular experimental model, that symptom correlates with the clinical condition. This is the meaning of face validity of experimental model. Second similar type of thing we have to decide, and that is construct validity. The construct validity means there should be similar underlying biology or pathology in that experimental model. Again, let's take example of anxiety. So when a person is suffering from anxiety, there is some problem in his serotonin concentration in his brain, or there are some other neurotransmitters which are altered in particular pathways. So if you are saying that a particular experimental model is a model of anxiety with construction validity or construct validity, then in that animal model, in the animal's brain, similar kind of changes must happen which happen in a depressed patient or patient suffering from anxious pain. If such kind of molecular basis is there, pathological process is similar, then we can call that model as construct or a model with construct validity. Third thing we have to look at is predictive validity. In predictive validity, we are talking about the ability of that experimental model, sensitivity of that experimental model to exactly show whether our drug is effective or not. And that model should be responsive to the therapeutic intervention that we are doing to that model. Say, again, let's talk about anxiety. We are saying elevated plus is a model of anxiety. Then, in that model, preoxetine at 10 mg per kg should be able to suppress that anxiety in the animals. If this happens, then we can say that our model is having predictive validity also. Then there is one more type of validity which comes in picture, which is called as etiological validity. This becomes slightly tricky now when we are talking about behavioral experiments. This kind of etiological validity becomes quite problematic. It will be very daring statement to say that an animal model will be having etiological validity in terms of psychiatric disorders. Because see what happens. Again, let's consider anxiety. In human beings, anxiety may come because of say examination. Right? Many students suffer from anxiety when they are going to appear for examination. Such kind of anxieties will never come in animals, or will not be able to model that examination related anxiety in animals. Or phobias now particularly. Phobias also, human being is having hundreds of different types of phobias. So we cannot create models for those phobias. So etiologically valid behavioral model is really very, very challenging because you cannot create similar conditions for that animal which are creating diseases in human beings. So do you understand now why validation of behavioral related models is more complex? Not only this, there are other aspects of validity where we are talking about convergent validity and divergent validity of the model. This all appears like a discussion related to quality control. But it is important that we should apply these principles to our animal experiments also. So when we are talking about convergent validity and divergent validity, we have to look at the meaning of these two terms. Okay? I hope there are pharmacology teachers and students sitting in front of me. So we have seen about elevated plasmids or marble burning paradigm. Marble burning paradigm is what? If you put shiny marbles in the cage of a rat or mouse, they show tendency of burying those shiny marbles. And this we consider as a sign of anxiety. Now the same experiment you will see as a model of depression. The same experiment of marble burying paradigm you will be seeing as a model of obsessive compulsive disorder. So what is happening here? The model of marble burying paradigm is showing same effect in depression, anxiety and obsessive compulsive disorder. So if suppose a model is working on multiple types of drugs and giving similar response, then we will be saying that that model is having problem of convergent validity. Means multiple drugs are showing similar effect in that particular experimental model. And then there should be something called as divergent validity. Divergent validity means what? Our experimental model should be able to differentiate between different classes of drugs. For this, we can take example of Cook's polar apparatus. Okay? In the Cook's polar apparatus, what we are having a chamber which is having a mesh, a metal mesh at the bottom, and there is a hanging central pole on which animal can climb and escape from the shock which is being delivered through that bottom mesh. Okay? So this is the simple construction of Cook's polar apparatus. And we all know that this Cook's polar apparatus we are using for testing antipsychotic activity of the drugs. conditioned response in the animals, or inhibition of conditioned response in the animals. If we consider this particular experimental model of Hooke's pull apparatus, then using Hooke's pull apparatus, we can differentiate between antipsychotic agents and sedative hypnotic agents. The kind of response which we get in Hooke's pull apparatus for antipsychotics is it selectively inhibits conditioned response. But that model differentiates the sedatives from antipsychotics, where sedatives will be suppressing both conditioned and unconditioned response. So here, our experimental model is able to specify or differentiate between the two different classes of pharmacological agents. So we can say that our model is having divergent validity also. So if you are having multiple anxiolytics like serotonin reuptake inhibitors, diazepam, or other agents, then all of them should show similar activity in the model. Then it will be called as a model that is having convergent validity. And if the experimental model is able to differentiate between different classes of drugs, then we will be able to say that that particular model is having divergent validity also. Now, beyond this, people have started to talk about ecological validity. This is again a complex term. Ecological validity means now we are saying that even the environmental conditions of that model should be similar to the clinical conditions. This is a distinction which cannot be achieved. But still, when we are talking about experimental models, animal studies, or any pharmacological model, we have to have such deep thinking about validity of that model in terms of etiology, in terms of symptoms, in terms of relativity, in terms of its ability to differentiate between the two different classes of drugs, its ability to show similar responses for the different classes of same type of agent. Anxiolytics, multiple classes show similar activity. If all these things are happening, then we can consider or we can say that our model is having validity to be a correct experimental model. Now you can imagine how difficult it will be to do such things in behavioral testings. In behavioral testing, having all this kind of validities is really very difficult. And that's where our experimentation becomes more and more tricky. Okay? Let's try to look at one kind of disease in human beings, which is called as anxiety disorder. What happens in anxiety is the person is having continuous fear of something, and that continuous fear is not having any proper basis. It is a pathological fear, and the fear is to such an extent that it starts interfering with the person's normal function. Now, if such kind of fear, which is for a prolonged time without any external stimulus, and which is affecting the physiological and normal functions of a human being, we call that this is just anxiety disorder. In anxiety disorder, again, there are multiple types of anxieties. One is called a generalized anxiety disorder. Second is panic disorder. Panic disorder is the disorder which comes with or without agoraphobia. See the complexities of psychological disorders here. Agoraphobia is the condition in which person is having fear of closed compartments. If suppose a person goes in a lift, then that closed compartment of the lift creates phobia in his mind, fear in his mind. So such kind of disease also needs a treatment. So panic disorder is a kind of anxiety. Apart from this, there are multiple types of phobias. If you search on any internet tool, you will see that hundreds of types of phobias are there. Someone may be afraid of lightning, someone will be afraid of water, gnats, then lizards, dogs, horses, sitting in a flight, speaking in front of a group, All these kind of phobias may happen in people. Okay? Now you can imagine whether we can create model for each of these phobias. It is impossible. Then there is social phobia, means people are not having daring or not having, or they become very nervous when they have to talk in a group of people. Beyond this, anxiety, a part of anxiety now, it's called as a separate disorder now, it's obsessive compulsive disorder. So here also, a person is obsessed about, say, cleanliness or obsessed about security. These kind of obsessions are there, which also come under anxiety disorder. And then there are post-traumatic disorders. Suppose a person comes across some bad conditions or bad experiences, then he develops PTSD. So, if a simple disorder of anxiety is so complex, is it really possible to create a behavioral model for this? Can we say that there will be a simple general model to tell about the effect of drugs on anxiety? It is not possible. So that's what we are going to look at now. When we say that we are having an animal model for testing anxiety, what we are saying? We are saying that that animal model is etiologically similar to human clinical anxiety. That model is creating similar symptoms as human clinical anxiety. And most important is that model will be sensitive to the treatment which is used clinically in human beings. So suppose we are having fluoxetine as treatment of anxiety, fluoxetine must show efficacy in reducing anxiety-related symptoms in that experimental model. All these things are happening. Then we can see that our model is a valid model for testing the behavioral disorder called anxiety. How far this is possible? Can we really induce continuous anxiety in animals? It is not possible, right? Anxiety for a prolonged time, which affects normal activities of the human being, cannot be replicated as such in animals. So, for the model that we are using for anxiety, we have to use a different word now. See, streptozotocin induced diabetes is an animal model of diabetes, human diabetes. Right? We cannot have a parallel to this in behavioral conditions. We cannot have a model of anxiety in animals because pathologies are different. In human beings, anxiety happens because of thousands of different reasons. Animals, those reasons are totally irrelevant. A rat will never feel that he is worthless. Okay. So the kind of anxiety we are projecting in this animal model of anxiety will not be a real anxiety. Rather, it will be a taste of anxiety, not an animal model. Can you get this difference, please? Streptozotocin induced diabetes is the animal model of human diabetes. But we cannot have a parallel kind of thing in case of behavioral disorders. Because for the conditions like anxiety, whatever causes are there in the human being, similar causes will never be there in the rat. That is the reason why we have to use the word animal test when we are talking about behavioral disorders. So such subtle differences we do not generally discuss during the routine academic classes. But if we have to look at the depth of really designing an experimental model, then we have to look at all these aspects. Here we are talking about whether it is a test or whether it is a model. Okay? So for behavioral disorders, in majority of the cases, we are having just animal tests for checking the activity. We are not having animal models as such of the behavioral disorders. Okay? Because what is considered is fear can arise from pathological processes which are upstream to fear response. What is meant by this particular statement? We may see some fear in the animals, but that fear doesn't represent human anxiety. Because for human anxiety, there are many pathological processes which are happening in human brain, and because of which the consistent anxiety is happening in humans. So I hope I have made these 2-3 points clear. First thing is, creating a model for behavioral disorders is very difficult. Validating a model for behavioral disorders is still more difficult. And third thing is, as these things are difficult, instead of calling that particular model as animal model, we should call it as animal test for checking behavior than an effect of drug on that particular behavior. So this is how people are looking at animal testing related to behavioral disorders. Scientifically, this is how we have to look at behavioral disorders, and we have to say that we are actually having test for checking the behavioral disorder in animal. It is not a model of disease. There is one more thing now. Difference between trait anxiety and state anxiety. So, for a simple disease like anxiety, we have to look at all these kind of complexities. Trait anxiety is what? Trait anxiety is actual disease that happens in human beings where person is continuously anxious without any external stimuli, and this anxiety is affecting his physiological functions or normal activities. That we call as trait anxiety. It may have genetic reasons, it may have some changes in the brain, it may have some correlation with the consumption of some substances and all. Opposed to this is state anxiety. If suppose you are standing on a very tall building and looking downside, there also you feel fear, right? But that fear is not a disease. Animals also have similar kind of fear. If suppose you make a ladder, you stand at a heightened platform which is very narrow, then that rat will also generate fear, its mind will generate fear. But that fear of height will not correlate with the disease anxiety in human beings. It will be a state related fear. As the animal has kept at a heightened surface, it is afraid of falling off, so it is showing some peculiar behavior, and that is behavior of state anxiety. So, whatever models we are having, they are related to state anxiety. They are not related to trait anxiety, which is there in the human beings. Okay, so let's go at this particular article and let's go ahead. Now, to model this particular condition, what we have done, we have developed elevated plus maze. Across the years, across the decades, many scientists have tried different kinds of mazes, and in 1984, for the first time, Henry and Mithani projected that we can have elevated plus maze like this, which will be having open and closed compartments. There will be a central compartment. And what scientists proposed is, as this maze is having height from the floor, this rat will be, or mouse will be afraid of the height, and it will avoid coming on this open arc. See what we are trying to model here? We are trying to model fear in the animal. What kind of fear? It is a state fear. We have not done anything or any manipulations in the rat's physiology or in its brain. Just its normal fear we are trying to look at. And this height is providing us with an opportunity, height of the open arm is providing an opportunity to see whether the animal is really afraid or not. And the closed compartment will be provided in a safer place to stay inside and feel free. Okay. So, if suppose our animal is anxious, it is fearful, then it will be staying more inside this closed compartment. If it is not afraid, then it will be coming on the open compartment. This is the simple principle on which elevated plus maze was developed, validated by people. Okay. For the state anxiety as a test related to anxiety, not a model related to anxiety. Okay. Now let's have a look at how actually this particular test is performed. In this particular model, I just told you that height of this particular open arm is more important, and the rat will be afraid of falling off. So, what we are looking at in this particular condition, we are looking at fear of height in the animal. But only not this thing. In animals, there is a property called as chemotaxis, which they always move across the wall. Okay? Even this may be a part of this particular behavior that we see on elevated plus. Then there are some visual cues about which the animals may be afraid of. And then they are having mustaches. Even male rat or mouse, they are having mustache. A very long hair are there. So those long hair also may play some role in animals' behavior on such elevated plusness. So all these things we have to look at when we are talking about elevated plusness as a model. And then what we actually record in this model is all these parameters: time spent in open arm, how many times the animal is coming in open arms, how many times it is showing freezing, how many times it is remaining in the central compartment, which we consider as a sign of confusion, whether to go in closed or whether to go in open compartment. This is the confusion that animals are facing. So all these parameters we are observing. Along with that, we also check many more parameters, like whether it is passing urine when it's coming on the open arm or not, whether it is going back or not, whether it is exploring that particular maze by standing up on the hind legs or not. All these parameters we are observing as ecological parameter in this particular model. I'm sorry that I'm going slightly faster because I have covered little more details about this particular experimental aspect. But what I wanted to tell you from these 3-4 slides is just to look at the state of fear in the animals. This elevated plusness model has been created, and we are checking effect of drugs in this behavioral test to see whether the animal's fear of height is reduced or not. And fortunately, all the drugs which have anxiolytic activity, they reduce this fear of height in the rats or mice, and the rat or mice starts coming on the open arm. And so the model is, so the test is used for checking anxiolytic activity of the drugs. Let's have a look at how this particular test is performed in standard conditions. And then we'll be looking at variations which are there. Okay? I hope this video is visible to all. Can anyone confirm, please?
Yes, yes.
Okay. So rapidly I am trying to show you how in a Japanese lab this particular elevated maze has been set. So we are having open arm here, closed arm here. Beyond that, you can see a balloon has been hanging. There is a reverse pyramid hanging on this wall. So let's remember these two figures now. Let's go ahead. Then the experimenter is looking at illumination which is available in the room. And the experimenter is setting the illumination exactly at 100 lux. This is light intensity. Okay? So if you really want to perform elevated plusness test in your lab, first thing you have to see that you will be having some visual cues like balloons or inverted triangles and something like that. Beyond that, you will have to look at the light intensity which is present in that room. Then, when you are bringing the animals in the experimentation room, you have to keep the cages in the lower racks. Why? Because exposure of these animals to brighter light may change their behavior again. Okay? Let's go ahead. When this test is really performed and finished, you have to wipe the entire elevated plusness with alcohol. See what the experimenter is doing here is, she is replacing the total arms, closed open arms of this particular elevated plusness, and she is wiping it with ethanol or isopropyl alcohol, 70% isopropyl alcohol. Why? To prevent olfactory cues. So what we are seeing in this particular video is scientists are taking care of whatever spatial arrangements are there in the experimentation room. That room should be totally sound free. Third is that illumination which is available in that room, light intensity which is available in that room should be specified, and at that particular light intensity only we have to perform the experimentation. We have to see that the animals are not exposed to deviating light intensities when they are being brought into the animal house. Beyond that, we are trying to remove all olfactory means smell related cues about the first animal that has been tested, and then subsequently the animals will be affected by that. Even olfactory cues we are trying to remove from alerted plasmids. Okay. Why I am trying to show you all these things is because these are the things, simple things, which are going to affect the outcome of our research. And let's see scientific testings where these things have been shown in more details now. Just now we had seen that there is a hanging balloon and hanging pyramid in this room. Similar kind of arrangements are there for all the behavioral tests now. Are we concerned about these environmental aspects? Do we feel that the rat moving on this maze will be looking at these things? Yes, it looks at those things, it remembers them, it learns which particular side of the room or which particular side of the alerted plus maze is safer for it. Okay? And that learning within 10 minutes is enough stronger, enough confirmatory for that animal, so that if you subsequently, if for the next day, if you use the same animal on this maze, it will be showing lesser effect to your tested substance. To this extent, animals' behavior is complex. And this particular response we call as one trial tolerance. Means what? If an alerted plus maze, if you test the animal for 10 minutes, then that animal you cannot test again the next day, the next week, or after 2 weeks. You have to wait at least for 3 weeks till the animal forgets about the ambience in that room. If you use an animal repeatedly on the alerted plus maze, then it will not be giving correct response to your therapeutic interventions or test drug interventions. To this extent, environmental ambience means whatever things you are putting in the experimental room, they are having an impact on the animal's behavior. And this has been very systematically tested, and you will be getting hundreds of such articles where one trial tolerance has been shown to affect the outcome of the experiment. Okay? That is the reason why the place where we are working, the place where we are observing the behaviors of animals, is very much important. Okay? It has been said that if suppose we repeatedly use the same animal on this elevated plus maze, then the animal's response to anxiolytic activity of diazepam will not be visible in subsequent tests. Can you imagine this? This kind of research has been published that if repeatedly you are exposing the animal to alerted plus maze, it will not be responding to diazepam later on. It will not be showing anxiolytic activity of diazepam in subsequent tests. But it will be effect, or it will be showing activity of preoxetine like drugs even in subsequent testing. From this, scientists have drawn very important inference that diazepam, as it is ineffective in subsequent testings on alerted plus maze, it is lesser effective against the phobic disorders. So if suppose someone is suffering from phobia, low dose diazepam cannot be the answer to that particular condition. There will be choosing some other kind of drug, other anxiolytics. So can you understand the importance of keeping the environment in a specific manner and avoiding repeated tests of the maze is having such a significant conclusion in case of alerted plus maze. This simple behavioral test, we can just look at our laboratory now and how we are performing this test. There is a batch of 50 students. There is only one single small maze. It has been kept in the lab as such. Students are standing around that particular maze. They are putting the animal in the maze. All of them are talking and all, and then we are performing the test, which really is a wrong way of performing at least alerted related test. Because we are losing many such important aspects of the outcome if we are not performing or we are not keeping the ambience properly for the particular test. And this is true for many other tests also. Many other behavioral tests need such kind of sensitivity about what kind of objects we have placed in the experimentation. No doubt we are having for learning and memory, we are having a test called as novel object recognition test. Because the animals have tendency to remember their environment. That is the reason why we have to be very sure and very assured that we are using proper environment. I told you about the light intensity. In case of experimentation related to behavior, we have to maintain light intensity to a fixed level, fixed intensity. And then we have to validate our models. Otherwise, we will be getting wrong results on that. Such many research articles have been published where light intensity has been shown to affect animals' behavior on the mazes. We are totally insensitive about these things. While performing behavioral testing, we have to even look at the light intensity which is present in the room of experimentation. That to soundproof room, which is having specific kind of special arrangement of the objects. All these things are going to affect our outcomes. Okay? So, effect of anxiolytics led to serotonergic nervous systems are affected by changes in illumination. You can imagine now, sometimes what happens is, in our laboratories when we test mg per kg fluoxetine, students come to us after the experiment that they need things. Even though they are using electric plasmas, they are having all these aspects. But if suppose light intensity has not been taken care of, then we will not be able to see effect of fluoxetine and anxiolytic in that particular test now, electric plasmas test. So I hope I am highlighting how environmental conditions are affecting our result outcomes. Third thing is, we cannot believe that all the animal species will be having similar responses, behavioral responses. Within a species, different states are having different sensitivity to the or different behavioral traits. In different ways, their behavioral traits may be seen. Okay? And you will find research articles where people have tried to figure out which animal is more sensitive to height, the height of platform on which it has been kept. Similarly, people have tried to find out which strain amongst the mice is having resistant to elevated plusness. Beyond that, if suppose you are individually housing the animals, even such housing can affect the outcomes of research, outcome of your test. Okay? Because even mixing animals from different cages on the day of experimentation may seriously affect their behavior. Okay? Because they are going into the group which is unfamiliar to them. And that may affect the outcome of a behavioral test. To this extent, we have to remain sensitive. Whenever we are performing any behavioral related test, we have to at least have this sensitivity that all the things that we are doing to our animals are going to affect the outcome. Okay? So handling, this is very interesting aspect now. The way to handle animals, starting from their birth to the day of experimentation, their handling, the way they are kept, the company in which they have been kept, everything affects the behavior of animal, at least on this elevated plus list, on which there is very systematic research available. So if suppose prenatally the animal is stressed, when it is in the mother's womb, if it is stressed, or if it is separated from the mother's in early days of its life, then the behavior of animal changes. Okay? If suppose you handle the animal 15 minutes per day with some love, then their response to behavioral test changes. I hope I can get 10 more minutes. Right? So please give me 10 more minutes, I will be finishing within 10 minutes. So what we are talking about here is effect of handling on the animals and their behavior. So very interesting study has been published recently in a high-impact journal. What they have done in this study, they have just conditioned the animals. Condition means they have just trained the animals to get tickled. या आपण गुदगुल्या करतो म्हणतो असं. That kind of tickling they have done in rats. And then they have seen that animals love this kind of tickling and they become conditioned to remain near the hand of the experimenter and if the experimenter wants to tickle them, they allow the experimenter to tickle. Now, a very beautiful aspect of this particular tickling is, as you tickle these animals, as you play with these animals, the chemistry, brain chemistry changes. The receptors related to serotonin get more expressed in the animals which are loving this kind of tickling. And such increased density of receptors has been proved in the brain of these animals which are conditioned to tickle. And authors have shown that this is what I am trying to tell you. See, on the left side, we are having a rat which is enjoying this tickling. Okay? On the right side, there is a rat which is reluctant to tickle. See the difference in their behavior. This rat is continuously remaining near the experimenter's hand and it is allowing it to tickle. But this right side rat, it is not at all liking this tickling. Okay? Now, this kind of behavior, this kind of handling, not only is affecting the behavior, but even it is affecting the chemistry in the brain, expression of receptors in the brain. See, the rat which is reluctant to tickling, not conditioned to tickling, is having lower density of oxytocin receptors. But the rat which is having affinity to human hand, it has been tickled by the human beings, has increased levels of receptors related to serotonin. To this extent, handling can affect animals' behavior. Okay? Beyond that, there are many aspects. Hormonal changes are there. The age is a factor because across age, testosterone goes on falling. When change in hormonal levels also affects animals' behavior, they are less sensitive. There is very nice study on bedding material which affects the animal behavior. There are multiple studies. You can search on internet that you will come across multiple studies. This one is the very interesting study. What they have done is they have seen how quantity of the bedding material affects animals' behavior. The quantity of bedding material is what? If suppose the bedding material is 5 millimeter thick, if the bedding material is 1 centimeter thick, if the bedding material is 3 centimeter thick, how it is affecting the animal's behavior, animal's physiology. That is this. Can we believe? We are just randomly putting some amount of husk in the animal's cage. But if suppose that husk is having different levels of height, then the thickness of the husk material also affects the animal's behavior. It affects animal's physiology, organ beds, body weight, length of the tail. Everything is affected by the thickness of bedding material which is provided to it. Okay? Similar studies are there where people have tried to look at effect of different bedding materials on the behavior of the animal. So, in this study, what this authors from Saudi Arabia have done is they have used different bedding materials derived from herbal substances. Okay? Whatever is produced in high quantity, they try to put the powder of it or flakes of it as a bedding material. And then they saw that there is vast difference in the animal's behavior if the bedding material is changed. Okay? We feel that what is there in the bedding material, we can put even papers or we can put a rice husk or sometimes you can even put some leaves or something like that, but that is not true. Even this bedding material is affecting seriously the way in which animals respond in experiments. Similar kind of study has been done by this another author where they have used wood shavings, wheat straw and rice straw as bedding material. And they have seen that all these three materials are having very distinct impact on the animal's behavior. They are responsive on different measures, open field arena, even rotor rod, all these things are affected by the bedding material that we are using. So we have to keep uniformity in the bedding material. Another important aspect in the animal house is quantity of ammonia. Okay? If your animal house is not properly ventilated and if ammonia is getting deposited, and to such ammonia, if suppose the pups are exposed, then throughout their life their cognitive process is affected. So we have to even be sincere about the quantity of amount of ammonia that is getting retained in the experimental room or in the animal house. So all these things are having severe impact on animals' behavior. There is one more interesting aspect: pheromones. All animals, they have tendency to secrete certain chemicals which provide hints about the surroundings, which provide clues about the conditions which are available in the experimental room. You will be amazed to know that there is a type of butterfly which secretes some pheromones, and another butterfly can understand or sense those pheromones from the distance of 1 kilometer. In butterflies, this kind of sensitivity to pheromones has been detected. So, impact of such pheromones means certain, say, gaseous substances or certain chemicals which are secreted by one animal in response to the environment, sensed by another animal which is subsequently coming into that environment. So, kind of pheromones also we have to remain very sensitive. It is possible that if you are having a predator's smell in the room, experimental room, it will be seriously affecting the behavior of animals on these behavioral tests. Similarly, now new research is coming up where gut microbiota also has been considered to affect the behavior of animals. That is the reason why we have to provide water, proper distilled water, or water that is potable, which is not having abnormal high count of fungi or bacteria. That kind of water provide. Otherwise, even the kind of water that you are providing to experimental animals significantly affects their response in case of behavioral tests. Okay. So, whatever we are doing with the animals, providing them feed, putting the bedding material, providing them with the water, keeping them in proper lighted conditions, all these things are going to affect the behavioral outcomes. So from these few things, I sorry that I have to go rapidly through this particular presentation, but these are the ways in which behavior of the animals is seriously affected due to the things that we are doing to them. And if we are not sensitive about this kind of impacts of environment on the animals, then maybe the response that they are giving to our interventions will not be scientifically acceptable. Even simple behavior like killing of the animals, just tapping on their heads, even that has significant impact on behavioral outcomes of the test. So we have to be very serious about all these aspects. If we really want to get uniform results, if we want to get scientifically valid results, then maintaining the animals in proper way, keeping all the ambience related conditions in a fixed level, all these things are very important in pharmacological assays. And when you are doing behavior related assays, they are still more important. Okay? So this is what I wanted to share with you. And I hope I have tried to convey the message that I wanted to give you. Animals now require some recreational facilities in the cages. And whatever I am showing you on screen, if this kind of recreational facilities are provided to animals, and if they are uniform for all the animals, then animals' response to drugs significantly changes. We lead towards more valid scientific results if our animals are really healthy, maintained in proper conditions, and provided with best atmosphere to them. Okay? So let's try to take care of all these aspects when we are thinking about the behavioral experiment. Thank you for this opportunity.
Thank you so much, sir, for valuable deliberations on effect of environmental settings on behavioral outcomes. It was a very informative session. I request to Dr. Abhinandan Patil, sir, to kindly present the session summary and give gratitude thankful note to the Dr. C.R. Patil, sir.
Respected dignitaries, so sir, it was very wonderful talk from your side related to the environmental enrichment and behavioral pattern. The most important aspect that you have talked related to the psychiatric disorders along with the validations, which is important for the correlations factor, which can include the clinical condition, biological response, reproducibility, predictive analysis. So wholly it was related to the enrichment model, which was the actual theme of our 2 days workshop. And we are fortunate enough that whatever the demonstration you mentioned over there, they're related to the elevated plus maze. So Dr. Sandeep sir is here, so we are going to have a live demonstration related to that. So last part that I want to mention over here, you just shared one of the publication from the self-press current biology, that is the tickling and bedding, which plays a very important role in the case of the animal grooming or simultaneously the experimentation which is needed. So really such small thing aspect plays a very important impact on the results. So that is been understood by all of us. So thank you sir for being online. So actually we were expecting your actual presence over here, but in coming days we'll believe that you'll be the part of this face-to-face interaction. So thank you once again for being the part. Thank you sir.
Now I request to puzzle volunteers to start the session.
The first question is the elevated plus maze measures. I repeat the question. The elevated plus maze measures.
Just do you want to know which kind of measurements we are taking on that? Hello?
Yes sir, please.
I'm not clear what is the question is. Do you want to know what are the parameters which we are counting on this elevated plus maze?
Actually sir, these are the puzzle questions designed for the students so that they can interact with whatever the part you are taught in the session. So those students who are going to interact and give the proper answers, we are giving them some sort of gifts as a part of token of appreciation.
I thought it was question for me.
No, that was wrong answer.
If you have 2 wrong answers, go for the next question.
The next question is, what is meant by the construct validity of an animal model? I'll repeat the question. What is meant by the construct validity of an animal model? Yes? Repeat it. No, it's incorrect. Anyone else? Yes? Yes, that's correct. Please come forward. Please introduce yourself.
Hello everyone, my name is Valerie Dillon. I'm a master student here at CI Alcohol.
Now I request to the Abhinandan Patil sir to handle the prize. Thank you so much sir. Thank you. Can we move for the next session? Wait sir. Sorry. I request to Dr. Yashwant sir to kindly felicitate today's chairperson Dr. Abhinandan Patil sir. Thank you so much. Now we have the lunch break, delicious lunch break, and after that we'll proceed for the next session of the Dr. Sandeep Patil sir, and then we have practical sessions again. on experimental animal handling as well as bone marrow aspiration. So I request to all delegates stay tuned with the sharp time at 2:30.
15 मिनिट
of Tripura.
टेक्निकल सर आहेत त्यांना सांगा त्यांना किती कमी त्यांना ते ॲडजस्ट करू
कॉम्प्लिकेटेड आहे. अरे कॉम्प्लिकेटेड आहे रे. लॅब मोकळी उड्या मारायला पाहिजे. गबस लॉक लावून लॉक निघेल त्यांना जर सांगत काय काय नक्की बाहेर येतं काढायला असू दे असंच आहे. या बाहेर
एक्सप्लेन पण करणार मॅडम केलं
Contributions have not only advanced in the pharmaceutical science but also in contributions have not only advanced in
and what we use a process validation itself which comes with value based for
हां बोला की सांगितलेच सूर हडपायचं झालं काय परवा तुम्ही स्क्रॅप कुठनं काढली ते कुठे ठेवला की कोपऱ्यातच आहेत ते काळ्या पट्ट्यात ती
Good afternoon, Leo. We consider you had enjoyed the food. Now we will proceed for our next session. It gives us great pleasure to welcome Dr. Sandeep Patil sir as our speaker. Please see, dear audience, the delegate and audience, please have your seats. We will begin the session. It gives us great pleasure to welcome Dr. Sandeep Patil sir as our speaker. So please occupy the dais. The session will be chaired by Dr. Poonam Chawla ma'am. I request Dr. Sonali, Deputy ma'am, to kindly introduce the speaker.
Very good afternoon, all of you. It gives me a great pleasure to introduce Dr. Sandeep Patil sir. He is a professor and head of the Department of Pharmacology, Dr. Shivaji Rao Kadam, College of Pharmacy, Kasbe Digraj, Sangli. Dr. Patil is a distinguished academician, researcher with a strong expertise in experimental pharmacology and research methodology. He holds PhD from the Shivaji University, Kolhapur and has made significant contribution to teaching, research and academic mentoring. He is also an approved PhD guide and co-guide at national and international universities and has successfully guided several postgraduate and doctoral students. On the personal note, it is a pleasure to share that Dr. Sandeep Patil is my senior from Bharatiya Vidyapit College of Pharmacy, Kolhapur, and his academic journey has been a source of inspiration for all of us. He is the founder of Biosite Institute of Research and Development, Sangli, established to promote quality research among students and young scientists. With over 120 research publications, multiple patents, and several books to his credit, his work reflects strong commitment to innovation and translational research. We are honored to have him here today. Thank you, sir. Thank you.
Okay, good afternoon all. Thank you very much for giving me such a wonderful opportunity to express my views in front of you. Thank you very much to the DY Patil Society, Principal Sir, Organizing Committee. Today's session is, we are talking about that the models of engagement from classroom theory to field conservation. Okay. So as a speaker, it is very difficult task to me that before lunch, if I take the lecture, again it is problematic because everybody is hungry. And after lecture, again it is also difficult because the whole blood goes to the digestion, not to the brain. So I will try my best to concentrate all of you here to my lecture. So again, one more thing, all of you are eager to see the live demonstration of the various models. So I have wind up my projects very short period of time. Okay. So animal testing. So in that animal testing, we are using the various animals, that's a rat, mice, rabbit, guinea pig, zebrafish, daphnia. So all these animals we are using for the experimental purpose to get the something new product that can be useful for the society, that can be useful for the various things. So in that animal testing, so we are using the various models are there. So that models we are performing in our college levels for the various activities. So today I will show you some models that can be used for in your college if you are performing the various activities. Okay, so in that things, so there are different animals are used in research, that is all of you know very well. So these animals are used for a different purposes. So these animals are basically 4 categories, in which first that is the experimental animals. So where we can induce the disease to the animals and then we perform the activity. Is it clear? Induced animals. Second category of the animal used in the research, that is the spontaneous natural models like as a EPM, angiolitis. We cannot induce the anything, just put the animals and just see the natural behavior of that. Negative, where we cannot give anything to the animal, just we are providing only the normal saline, that is called as a negative control. And orphan model, so diseases which are there is no human analogs, for that purpose we are using the animals. Okay. So these are the different animals which are used in the laboratories. I hope all of you have seen in the animal house. So these 3 animals are also used in the research areas, that are fruit flies, zebrafish, worms, that is the sea elegans. Okay, so these are the common animals which are used in the research purposes. So today's our topic is a theory to the field. So that's why I have decided that we have taken some part of the theory. Some part of the theory is some drugs that can be repurposed, and how can we use the different animal models for to prove that activities. Okay, so sincerely I am thankful to the Goa University. So final year students, they have prepared the repurposed drug PPT, and I have just taken some few PPTs from that slide and explained the animal models for that purposes. So today we are going to cover some drugs that are all of you know very well common drug. There is aspirin, captopril, ondansetron, COX inhibitors. Okay, it is a painkiller, it is analgesic, antipyretic, all categories all of you know. So in the mechanism basically, if you see that the phospholipids which form the arachidonic acids with the help of enzyme, that is a COX enzyme, it will be formed to the prostaglandin G2 to prostaglandin H2, and these prostaglandin H2 which converts into the formation of different induces platelet aggregation, vasoconstrictor, means simply pains. They are formed the pains. Okay, so here the aspirin is well known for the COX inhibitors and which gives the pain relief. Now we are going to target, that is a P53, by using the various molecular softwares, devices, discovery softwares. In that we have to find out these aspirin can be targeted to the P53, that is a tumor suppressor pathway. So repurposed target of aspirin is the cancer therapy, in which breast, colorectal, lung and prostate cancer. Okay. How it is targeted as a cancer aspirin? Because so this aspirin which is the block the MD2 receptor, which is the normally degrades the P53. So what is our target? To restore the P53. So what is the role of P53? So that is a bind to the DNA proliferative thing and activate the tumor suppressor. Okay. So tumor suppressor activity is carried out by the cell cycle arrest, second apoptosis, and third that is the inhibition of the inflammation. Now I got aspirin, which is as a painkiller, but repurposed target is a P53, which acts as a cell cycle arrest, apoptosis and inflammation. Now I have to start to work on that. First, that is aspirin. It is how to prove that it is aspirin, it acts as a COX inhibitor. Okay, so in my college, that is one of the apparatus is there, that is a Hugo Bessel. That is the plethysmometer, digital plethysmometer. Okay, so by using the digital plethysmometer, I have to check the inflammation which is induced by the carrageenan. Okay, if carrageenan is not available, okay, no problem. Egg albumin is available. So by egg albumin, I can induce the inflammation into the rat paw. See here, there is a normal rat paw, and here is the inflamed rat paw. And second, previously 10 years ago, when Hugo Bessel is not available with us, at that time we are using this mercury manometer. So by mercury manometer, it is called as a again some colleagues said we have not animal house, but still we want to perform the anti-inflammatory. No problem, just go to the chicken slaughterhouse, just take the paw of that chicken and try it to check the anti-inflammatory activity by using the chicken legs and their paws. Okay, so by this way I can prove that, not scientifically, but just like as a anti-inflammatory activity by using the aspirin. Now my next target is a P53. So for that purpose I have to perform the anti-cancer activity. So anti-cancer activity perform, I require some, that is a media, DMEM, MEM, RPMI. So here 96 well plate is required. Here is inverted phase contrast microscope where we can see the growth of the cancerous cells. Okay, and here MCF breast cancer cell line. Okay, that photo was taken at the 2013 as during my PhD work. So I have seen the MCF growth, cell line growth. Now next target is my MTD assay. So aspirin I have to check whether it is a showing the activity or not. So for that purpose I have used the MTD assay. MTD, SRB, trivalent, crystal violet, neutral red. There are so many dyes are there that can be used for the anti-cancer activity. But this method is a valid all over the world. This method is a carried out. Okay. Now what is exact this MTD? So MTD is nothing but that is the, it is a dye. It is a dye which have yellow color, but when it is applied, come in contact with the cells, it starts to convert into purple color. Clear? Yellow color which converts into the purple. Why it is change color? Because of the metabolically active cells can only change the yellow color to purple color. Okay. If my drugs have killed all the cells, so definitely there is color changes reduces, and I can check the intensity of that color under the colorimeter or spectrophotometer or ELISA reader. Okay, so amount of formation, so whatever the formation is formed, that is produced is directly proportional to the cell number. So if I take the 10,000 cells, so I have to check how much cells have converted the yellow color to purple color. MTD assay. Next, so this is the procedure which is you can get everywhere. Apoptosis. So for that purpose I have used the dyes. There are so many dyes are there. Anisin dye is there. DAPI dye is there. So DAPI, which is bind to the nucleus and it is fluorescence. Under the fluorescence microscope I have to check the apoptosis study. Necrosis study, apoptosis. It is a programmed cell death. So I can play apoptosis by using the flow cytometry, and here I have to check that the aspirin, it is really shows the apoptosis by flow cytometry or not. Even that I have explained here in lab previous slides. That is the aspirin shows the inhibition of the inflammation and angiogenesis. Now for that target I have to choose the CAM assay. So in that CAM assay I have to procure the eggs. So here the point is there, I have so many times I have procured the eggs, but that is not properly hatched. So fresh eggs within 48 hours, if you place into the incubator properly, then and then they have produced the angiogenesis formation. So different eggs have to hatch into the proper way into the incubator, open the cap, and in the vessel you have to put the drug samples, whether your drug sample is a solid or liquid, and you have to check whether there is hemorrhages, coagulation or vessel ruptures. So on that basis, so my target aspirin as a repurposed in the cancer point of view, I have to check. Next drug, captopril. So all of you know very well captopril. It is a ACE inhibitor, angiotensin converting enzyme inhibitor. So theoretically, all of you know very well the mechanism of the captopril. So this captopril is a inhibits the which enzyme, that is a ACE, that is the angiotensin converting enzyme, and after binding to this enzyme, so angiotensin 2 causes the vasodilation, lower aldosterone and decreased blood pressure. So this is basic mechanism I have to know that. Now mechanistic hypothesis of this captopril. So captopril, which is the block to the enzyme that is LTA4 hydrolase. LTA4 hydrolase. So which is helps to the conversion of LTA4 to LTB4. So what is the role of LTB4 in the body? That is airway inflammation, recruiting neutrophils, hyperresponsiveness. Okay, and if I block this enzyme, so decrease the LTB4, so that can be decrease the neutrophil, decrease the vascular permeability, and decrease the inflammatory cytokine. So this will be helpful in the asthma as well as arthritis. Okay, so captopril as a repurposed, I can try to the check use in the asthma and arthritis. So captopril, it is a AC inhibitor, showed affinity toward the leukotriene A4 hydrolase, so indicating anti-inflammatory application in the asthma and arthritis. Now, my target is start to the prove that whether captopril is used really as an AC inhibitor. So I have to use the small X protocol. So where I can procure the chemical substrate HLL from the sigma and perform this experiment, and where I can use the captopril as a standard. So simultaneously you have to take the another drug samples to check whether it is really work as a captopril or not. So here captopril as a AC inhibitor, that I have to check firstly, and then I have to check this captopril or another drugs on the daphnia. Okay, so it is one of the wonderful species there. So nowadays I have to use this work for the cardiac study, cardioprotective. So there is no any ethical requirement, only just handling is required properly. So in this Daphnia magna, it is a very small species where we can check the cardiotoxicity, cardioprotective activity of any sample. Okay, so in that cardioprotectivity, the heart rate is around the 180. So we have to just only count for the 10 seconds and multiply it by 6. So you will get the 1 minute count of that heart of that Daphnia magna. And overall, you have to just add 1 drop of the ethanol, CNS, cardiac depression is occurs. Add just 1 drop of caffeine on that, it is shows the stimulant action. Okay, so it is one of the wonderful model is there in the cardioprotection study. So I just request to that there is one video is there in that folder. That is a how to see the Daphnia magna. So it is a very small species, and you have to just only the cavity slide, put in that cavity slide that Daphnia magna, and see there is the heart is pumping there. So that heart you have to count it, and you have to see that there about this. Yes. So this is the setup of the this is cavity slide. So this is the Daphnia magna where you can check the cardiotoxicity, cardioprotective drug samples. So here your captopril, their role AC inhibitors. One model I have performed, that is AC inhibitor in vitro. One model from the Daphnia magna. Now next, that is the arthritis. Whether it is a captopril showing the arthritis model or not. So for that purpose, asthmatic model you know very well in the laboratory level, go tracheal chain. Okay, so go tracheal chain, you have to take it, make the rings proper and put into the organ bar and put the acetylcholine, histamine, and check whether your sample tentatively whether it shows the anti-asthmatic or not. Guinea pigs. Guinea pig, it is very sensitive animal for the histamine, acetylcholine. So if you tap more your feet in front of the guinea pig, immediately heart attack is occurs for the guinea pig. So very sensitive, very what do we say, that is a useful animal in this study in asthmatics. So here we have to use the aerosol in the histamine chamber, and we have used the acetylcholine or histamine for the bronchoconstriction and measure the time for the convergence. So here we have to measure the pre-convulsive dyspnea, difficulty in the breathing of the animal, and immediately you have to remove the animal from that side if there is a convergence occurs. So how much time required your drug sample to extend the convergence or pre-convergent formation? If such a model is not available, then another model is the milk induced. Simply there is a boil the milk cool down that milk and just use the 1 ml milk, 0.5 ml milk subcutaneously to the animals, to the mice. So there is the increase the eosinophil, leukocytosis. And in another animal groups, you have to give your drug sample and check the level of the CBC, that is complete blood count. Clonidine induced master cell granulation. So by this way, captopril can be checked as an anti-asthmatic and simultaneously arthritis. So there are also in vitro, in vivo models are there. In vitro model, that is the protein denaturation model is there. Okay, membrane stabilization model is there. Proteinase model is there. So in vivo, there is the CFA, complete France adjuvants. So that is nothing but that is the dead Mycobacterium tuberculosis. So that can be used and perform the collagen induced arthritis. So for that purpose, we have to perform the model, that is the X-ray. So recently we have to use the X-ray. Dental X-ray can be for the check the arthritis inflammation. Second, various biochemical parameters, that is SOD, catalase, GSH. Third one, histopathology. Fourth one, that is the using the vernier caliper to measure the size of that arthritis. So different parameters are there. So simultaneously you have to increase the different parameters to measure the anti-arthritic activity in the animal models. Okay. So these are indirectly whatever the repurposed of the captopril that can be I have to check in the anti-arthritic and anti-asthmatic models. Next, ondansetron. All of you know very well, ondansetron, it is nothing but that is the anti-emetic drugs. Okay. So these ondansetron, which is bind to the 5-HT3 receptor, which is ligand-gated ion channel, and that is located on the both centrally as well as the peripherally. In the peripherally on the vagal afferent nerves in the GI tracts. Okay. So these are triggering the afferent signals to the vomiting center, which is in the present in the medulla, and resulting in the nausea and vomiting. So our target is the identified, that is the 5-HT3, and we have to repurpose the target, that is the anti-Parkinson, Alzheimer, then schizophrenia. Why? Because such a receptors is also present there. So in the Parkinson, ondansetron, which are used by blocking the 5-HT3 receptor on the serotonin terminals and reduces the erratic and unregulated dopamine release, which is minimizing the L-dopa induced dyskinesia. Okay, so it also blocks the 5-HT3 mediated GABA release that would normally suppress the movement promoting neurons, and this improving the motor output. So this is the basic concept about the Parkinson disease, schizophrenia, Alzheimer disease. So this is the ondansetron probably blocked the 5-HT receptor, 5-HT3 receptor in schizophrenia, 5-HT3 receptor in the Alzheimer disease. So in the Alzheimer disease, there is increase the acetylcholine and increase the memory parameters. So what is my target is that how I can perform the different models related to the anti-Parkinson, schizophrenia and Alzheimer disease. So for that purpose, I have to use the model first, that is emesis. Okay. So ondansetron has a producer activity, that is the anti-emetic. So in that anti-emetic, I have to use the chick. Okay, a small chick which is given the copper sulfate, which is administered orally, and it starts to produce the riches. So that riches which is useful to check the whether your ondansetron has an anti-emetic activity or not. Once we have to complete that, now next my target is that is the Parkinson. So ondansetron, that is a haloperidol induced Parkinson disease in the mice. So for that purpose, I have to induction 1 mg per kg for 7 days. So haloperidol given for the 7 days, and after that I have to check the behavioral test, in which first that is a rotaron. So in Parkinson there is a problem, is the only basic word, that is the dyskinesia, means movement. So how can we have to check the movement? That is by using the rotaron. So movement impairment was indicated by the inability of animal to remain on the roller for the 3-minute test. So for the 3-minute test, we have to confirm that whether your sample, unknown sample, or whatever the ondansetron has produces such type of activity or not. Second, that is a catalepsy bar test. Here we have to check the average latency period. Average latency period, how much time the animal is hold their one leg on the block, or 2 leg on the block, or on the bar. So here the cutoff time is the 180 seconds. So these basic parameters you have to keep in your mind, and simultaneously you have to perform this experiment in the laboratory. That is a catalepsy, and it is correlated with the Parkinson disease. Anti-Alzheimer disease. So there are different mechanisms are there. So in which first mechanism, that is a anticholinesterase enzymes. So what is the role of anticholesterase enzyme? So we require acetylcholine. Okay, and what is the role of acetylcholinesterase? That is a breakdown this acetylcholine. Okay, if I block this acetylcholinesterase, the breakdown is not occurs and get the more acetylcholine, so get the memory enhancing. So for that purpose, I have to perform this experiment externally by using the acetylcholinesterase enzyme. Okay, now I have to procure the acetylcholinesterase from the sigma. It is very costly. I have to start to work on, and then I feel that why not we have to reduce the cost of this acetylcholinesterase enzyme. So I just got one reference, that is the chicken brain. So I have to procure the chicken brain, I have to centrifuge it, but still I am confusing that whatever I have isolated, that really acetylcholinesterase are different. So validation is most important, and once you have to validate whatever the supernatant which is obtained from the chicken ileum, chicken brain, that is acetylcholinesterase enzyme, then we can perform the lots of drugs in the Alzheimer activity as the acetylcholinesterase inhibitors. Second, that is an antioxidant. So in the antioxidant, we have to use the SY cell line. It is a neuroblastoma cell line, and here we have to induce the or damage to the cell line, neuroblastoma cell line, by using the hydrogen peroxide, H2O2, oxidative stress. And in against that, I have to check whether my compound is a protective or not, and for that, by this method, chronic oxidative exposures, I have to prove that that sample can be useful as an anti-enzyme. Third one, that is the anti-inflammatory. Here also, whatever the cell lines which is treated with the H2O2, okay, so that cell line can be centrifuged, and whatever the parameters are there, so in which that is a NFKB that can be checked by using the kits. Okay, by 3 different way models, ondansetron can be checked as an anti-Alzheimer or not under the repurposed condition. Okay, so Y-maze, 4-stream test, okay, novel object recognition, there are different models are used in the animals, and that in the animals, Alzheimer can be produced by the ALCL, that is the aluminum chloride. Okay, recently whenever visit to the any tea stall, I see that there is the aluminum chloride, what do we say that is? The tea which is prepared in the aluminum chloride, that tea, the tin. So I suggest that, so we use the aluminum chloride for the Alzheimer purpose, and you are preparing the tea in that tin, and that is provided the Alzheimer's to the all over the nation after 20 years to 30 years. So use the steel or another tin for the preparation of the tea. So in vitro models are there for the performing the anti-Alzheimer activities. Next, clofibrate is there. All of you know very well clofibrate. It is in the liver. It is converted into the fibric acid, and this fibric acid which is activate the receptor, nuclear receptor, that is a PPAR alpha. So PPAR alpha forms heterodimer with the RXR, that is a retinoid X receptor, and this complex which is enter into the DNA and perform the target to the genes and reduces the inflammation, reduces the triglyceride, increase the LDL particle size, increase the HDL synthesis, increase the cholesterol. So this is the basic mechanism in the pharmacology way we know very well that. Now my target is that type 2 diabetes mellitus, this receptor. So in that type 2 diabetes mellitus, so first I have to check the clofibrate as a reduction in the hyperlipidemia. Okay, so first I have to perform in vitro. So in vitro here we have to check the lipase inhibition assay. So here in previous slide here we should see there is the inhibitor of the lipase enzymes. So lipase enzymes can be performed by using the titration method. So just take the 7 ml oil emulsion where the 2.5 ml deionized water, 3 ml oil, olive oil, 1 ml is the tricessial vapor, and 0.5 ml detergent. So these are the prepared in one, and in the mixed with the your drug samples and place on the magnetic stirrer for the, and then add the lipase enzymes in that and incubate for the 60 minutes. And after that you have to stop with the reaction with the mixture of acetone and alcohol, and this reaction is titrated with the NaOH by using the phenolphthalein indicators. So by this way we have to taken the orlistats, it is a standard drug, and whatever the readings of the orlistat, that I have to compare with these drugs. Okay, and at the end I got that lipase inhibition assay. So similarly HMG-CoA reduction is also performed in vitro Similarly, co-fibrate-like drugs can also perform the anti-hyperlipidemia by using the high-fat diet induced Tritonex, Coloxamar 407 induced hyperlipidemia, 25% fructose induced hyperlipidemia. So there are different models are there. Okay, here the one important thing I have found that in the high-fat diet, I have prepared the diet so many times. I have prepared the handmade diet our students, and I have given to the animals. Animals are not ready to eat. Okay, because they need something different. So we have to add the lard from the slaughterhouse, and now they are start to eat more and more, and then it gets the weight gain is there. High-fat diet is on. Okay, so different times you have to perform the different experiment, different way. So then you will get the actual lacuna in that stories, actual problems in that procedures, and then sort out these issues. Diabetes models, all of you know very well, anti-diabetic model by using the streptozotocin. It is a well-known model. Second model that is the alloxan-induced, where high mortality is occur, and third one model that is a dexamethasone models. So insulin resistance purpose, a low cost model that is a dexamethasone 10 days model where we can perform the diabetes into the animals in the rat mice. Okay. Now in vitro. So in vitro there are so many models are available in the diabetes by using the cell line also there. Alpha amylase, alpha glucosidase, glucose uptake, glucose adsorption. Then in the cell lines we have to check that is a T3, there is the muscle cell lines here where we can check the glucose consumption, glucose uptake and GLUT4 expression studies. And here the clofibrate I have to check for the diabetes purpose whether it is a really shown activity or not. Linagliptin, it is one more drug that is a DPP4 inhibitors which is used in the type 2 diabetes, primary target is there. And here see the diagram, oral glucose which is enter into the intestine. So in the intestine there is the release the hormone that is incretin. Okay, so DPP4 which is the enzyme play important role to the degrade this or inactive this peptide. So if I use the inhibitors, so what happens, that can be decrease the glucose plasma and decrease the glucagon, and it acts indirectly as a anti-diabetic model. Now my target is that is after finding the various roles, I find that it is useful in the fibroids. Okay, so this is also bind to the fibroblast activation protein alpha. So uterine fibroids, it is nowadays problem is occurs, it is a non-cancerous tumors, and now I have to check whether it is really work out or not for that purpose. So this is the pathway that is linagliptin has shown the DPP inhibitor. So it is a decrease the fibrosis, inflammation, fibroid tissue stiffness. Okay, so in that basis here decrease the interleukin 6, TNF alpha, decrease the chronic inflammation. So my target is that how to I can prove this by using the simple model whether it is a linagliptin has shown the such type of effects or not. So for that purpose I have to use the one more model that is the Agrobacterium tuberculosis by using the potato. So on the potato in the agar, so I have used that bacteria, so that bacteria has produced or ability to produce the tumors. Okay, and if I use our drug samples and check how many tumors are formed on that potato, so on that basis I can say that this drug can be reduce the tumor. Okay, and ultimately not directly, not say I am 100%, but I can say that whatever the linagliptin has another target fibroids that can be reduces the tumor. Okay, so this is our website where you can WhatsApp logo is there where you can join. You will get the details about the conferences and all these things all over the India. Okay, thank you very much for giving me such a wonderful opportunity to express my views about this predictive model. And I am thankful to the Dr. Neeraj Viwari sir, who is the referee of my PhD, and their kind support is always there with us for doing the various biological activities here and there. And thankful to the principal Dr. Jangme sir, they always support to us to do something different and in the biological activity. And I am very much happy that all pharmacologists are here, and definitely in future definitely they will prepare the new models for the pharmacology. Thank you very much.
Thank you for the insightful session. Now I request Dr. Poonam Chogle ma'am to kindly summarize this session.
I felt truly privileged to chair this session of Dr. Sandeep Patil sir, as he is one of my mentors and is widely regarded as one of the best researchers in Western Maharashtra through Biosite Institution of Research and Development Center. He has provided a strong research platform for budding researchers. Today, he delivered a insightful lecture on the models of the engagement from classroom to fieldwork, which was both inspiring and highly enriching. In that, Sir has deeply emphasized on categorized animals, drugs, and their mechanism of action, for example, aspirin and their activities of the anti-inflammatory. Also delivered a key point on the anti-cancer activity by MTT assay. Also explained keenly on the apoptosis and their hypothesis. So, it was a wonderful session, sir, and it was very informative and knowledgeable for young researchers. It will definitely help in their field work. So I thank you very much for accepting our invitation as a speaker, and on behalf of our organizing team and Divided Family, I will give you the very thankful thank you.
I request Dr. Poonam Chogle ma'am to felicitate the speaker with mementos. I request Mrs. Samruddhi Patil ma'am to felicitate Dr. Poonam Chogle ma'am.
Dr.
Hello. Okay, today all of you are eagerly demonstration. So we are going to there to show the zebrafish model. Before that, it is a very difficult task to the. So I will show you the one video regarding about the how to oral dosing are given to the zebrafish, and rest experiment that I will explain there with the live demonstration.
We have with us Dr. Budkar. I request Dr. Neeraj sir to felicitate Budkar sir. Thank you, sir.
Now it's my honor to introduce Dr. Santosh Valvekar sir, who has completed his Bachelor of Veterinary Science and Husbandry and Master of Veterinary Sciences and professionally trained as a veterinary surgeon and radiologist from Mumbai. Dr. Valvekar sir currently working as a laboratory animal veterinarian in the Department of Pharmacology at D.Y. Patil Medical College, Kolhapur. Dr. Valvekar sir have been serving as a veterinary officer with the Kolhapur Forest Department since 2018. Furthermore, sir is the Chief Veterinarian at Valvekar Pet Care Clinic, Kolhapur. The Dr. Valvekar sir invented India's first silicon-based prosthetic flipper for olive rattle sea tortoise. With this, sir has great research work on diagnosis and treatment of teeth canal affections by thyloscopy instruments. With great respect and honor, I request Dr. Valvekar sir, Santosh Valvekar sir, to kindly place the live demonstration and relate with us with expert session. Thank you.
Hello, 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 hello,
Hello, hello. Hello, hello, hello.
Hello. Hmm.
Hello again. Today we will be seeing a demonstration of another apparatus which is called as rotarod apparatus. So here you can see, first we will be seeing a short instrumentation, the basic objectives of the rotarod apparatus. We are basically using this apparatus to examine the skeletal muscle relaxant activity or the motor coordination activities of the drugs. So if we are going to administer the CNS depressants or the drugs which are going to cause the motor in coordination into the animals, that can be tested using the rotarod apparatus by a single parameter, that for a longer period of time, if the animal is able to stay on this rotating rod, then it indicates that it is having good grip strength into the muscles. And if it is not able to do that, that means it is having some of the skeletal muscle relaxants activity into the drug that we have administered to it priorly.
Now we look here, instrumentation. Okay. So when we see the instrumentation here, so one by one we'll see in the back side of the instrument there is a one cord which supply the current to the instrument, and one fused button is there backside the instrument. If you see in the front of this instrument, you will find here main button. When we start this main button, it supply the current to the electrical assembly. Then second button, which is the start button, when we press the start button, here timer will be start. This is a fortunate, or this is a benefit of this instrument, it not require the stopwatch, because already displays there, which is act as a timer. When we start the button, timer will start. These are dedicated for this first compartment. Second timer is dedicated for second compartment, and third timer is dedicated for third compartment. If you look this rotor apparatus, so there are three compartment model: first, second, third. Okay? First, second, third. So if there are different type of rotor apparatuses there, three compartment, five compartment, four compartment. So here at present we have a three compartment rotor apparatus. Okay? Benefit of this instrument, we can evaluate the drug activity in the animal in three mice or three rat at a time. One mice can be keep over this rod, second on second chamber, third on third chamber. In this way, we can check three animal at a glance. So this is the benefit of this assembly. We can check or evaluate the effect of the drug at a glance for three animal. Now we go to another part, that is the rod. Okay, this rod, if you see the rod, there is a fine grips is available on this rod, which help to handle the rod by the animals. When animal keep over this rod, animal try to hold this rod by this gripping and try to protect itself to prevent fall down this area, right? So this is a space particularly between the rod and autocut plate. So this, if you see this rotor rod, it's having the unit, that is the RPM. RPM full form is the rotate per minute. Okay. If how you can check its RPM? Suppose you mark here and see ki, after one round is complete, say ki is a one round. So we say ki how many rounds is completed. So we check per minute how many rounds are completed. If in 1 minute 20 rounds is completed, we say the 20 cycle is completed and it's 20 RPM. If it completed 25 RPM, so we say the 25 cycle is completed. But fortunately, or benefit of this digital instrument, we have a display. We can say what is the cycles of this rotation or RPM. So here we have a 11 RPM. So this is the benefit of this assembly. Everything is given. Otherwise, in traditional assembly, we require to check. Then, madam.
Also along with that, we also have a knob for setting the rotation, that is RPM. If we do want to increase the RPM, now you can see from the camera, I think, that the rod is moving more faster than the previous, and we can also decrease the speed accordingly. Now this may be required into various type of experiments, that we can increase or decrease the difficulty level of the rotation of the rod, which will increase or decrease the difficulty level for the animals to hold the grip onto the rod. So, and the second one here is the shaft, which is the rotating, which controls the whole rotating rod. Therefore, this basically the apparatus is called as rotating rotor rod apparatus, because the rod is continuously rotating on which the animal will be placed. And before doing the main experiment, we will have to train them prior to 3 to 4 days, which will make them understand that the rod is going to rotate and they have to hold a proper grip on that. And if prior to the experiment we administer them with any kind of muscle relaxant drug, then the grip, the time, the latency time after which they will fall, that will decrease, and the decrease in latency time will indicate that the drug which we have administered is having a muscle relaxant activity. And one more thing, when we are going to place the animal, after the animal will fall onto this plate, the timer will be automatically stopped over here and we can measure that.
See, if we see the next part, when we start the button, this display will be started. Okay, timer will be started. Now here, when animal keep over on the rod, animal try to hold this rod continuously. When it fall down on the auto cut plate, this is the auto cut plate. When it fall down on the auto cut plate, this will come down in such a way, and then there is a one minute button. Here is one minute button, and this is a very small button but very useful button, because when animal fall down on this plate, auto cut plate, then this button, which is the auto cut button, is also pressed, and then timer is stopped. So if you look this chamber, one chamber, so when animal fall down, animal will come on the auto cut plate. This auto cut plate connect to button, and button connect to timer. So let's see example. Just we start here. Okay, so timer is started. I have pressed the button start. Timer is started. This timer for the first compartment, the second timer for second compartment, third timer for third compartment. So when animal fall down, here reading will be there 13. When second animal which fall down, reading is 18. When third animal will fall down from rotor rod to auto cut plate, it will press the button which is below the auto cut plate. That button is pressed, and which connect to timer, and timer will also stop, and it shows the reading. What is the reading of animal of falling time? So it is a falling time of first animal 13, it is a falling time of second animal, it is a falling time of third animal. So in such a way we can note down the reading of the animal.
Now we will see the actual demonstration by using the animals. For this again we have taken 6 mice randomly divided into 2 groups like yesterday. Like the first group is the standard group, and we have just administered the distilled water to them, and the second group is test group in which we have administered a unknown drug to the mice. Now while doing the experiment we want you to note down the timing, and at last we want you to tell the answer, like what drug could have been administered to that mice and what activity you have seen into the test group as compared to that of the standard group. Just so according to the on the basis of the timing, you have to tell that the drug which was administered is muscle relaxant, muscle or the CNS stimulant or depressant, etc. So we will
Now
be
we
starting.
have seen the instrumentation, okay, how this instrument is work. Now we will see the demo, okay, how when animal is keep over on the rotor rod, and when it fall down, how reading is noted down. Okay. So first we will see the 3 animal of control group. Okay. See, timer is there. We will also noting down its time, and you also noted down the time when it's fall down. So this is a control group animal. Okay. So these are first. So this is the first animal, second animal, which try to hold the rod, third animal, and now we start the timer, okay? Timer is started, animal try to hold the rod, see, 1, 2, 3, animal try to hold the rod, timer is started, of each compartment, 1, 2, 3, and speed is 16. Actually recommended speed of RPM is 22, 25, but we are keeping here 17 to understanding how animal try to hold the rod, and for this we have given the practice to the animal. If you are doing the first day, so they may be fall down immediately or lose the muscle grip. So we have given the practice from last 7 days. So they are well trained animal holding the rod for prolonged time. Now we will increase the speed somewhat, just somewhat. But, yeah. So this is a control group. Actually, speeds will be same for control group and test group both, but we are doing here demo, so is alter. Increase the 20 speed. We are increasing the speed. Down. So I think this is the effect of practice. There is no fall down. Reading is continuously. Okay.
Okay, so we will be stopping this. We can note the time. Just stop.
So I think all you have noted, maximum reading is there for control groups. There is no any treatment. Only solvent was given to them. Now we will see the drug effect. Take an unknown drug. We will say the unknown drug, right? I will not saying which drug is given. You tell, is it CNS depressant or stimulator or what? Skeletal muscle relaxant or what property? First animal. Second animal and third animal. Now, madam, we will start. Timer is started. 1, 2, 3, and rod speed. Now, since 26 RPM is there. See, reading is second animal 33, right? Third animal reading is 37. First animal is well trained, but fall down, reading is 44. So, this is other reading of timer 1, means first animal, fall down reading, okay? 44, second is 33, third is 31. So, you may take the mean of this and see the test drug result, and similarly you can take the mean of control and you compare कि my drug is skeletal muscle relaxant property or not. If yes, ma'am.
So I hope you all of you have noted the time. I hope it was visible to you, and accordingly we will have to tell the results over there, and after that we will just conclude the practical that this apparatus could be used to determine the skeletal muscles apparatus for the rats also, and only the major principle on this on which this apparatus is working on the latency time. Latency time is nothing but the time after which the animal is going to fall onto this plate and the timer is going to stop. If this time is less than that of the standard group, that means our drug is going to be having a muscle relaxant activity, and if not, that means the unknown drug is not having any such kind of ability which is making the muscles relaxed. So this was the
if there is a muscle relaxant drug is there like benzodiazepine derivatives, diazepam. So diazepam act on the its receptor at the GABA associated receptor and then facilitate the GABA to act on the receptor. Then it open the chloride channel, chloride influx and causes CNS depression. Ultimately it causes a relaxant property. If in the second group we have given the diazepam, then it will relax the muscles. When muscles will relax, they are unable to gripping the rod. When they are unable to grip the rod, ultimately fall down. When they fall down, reading will be noted down. So it indicates that we have a skeletal muscle relaxant drug. So just for as a conclusion. Yeah. Thank you very much to all for listen carefully. Thank you. I have given the assessment to you all. Just attempt that. Most of the information also given in the MCQs and true and false assessment. Thank you very much all.
Thank you. Thank you all.
Hello. Hello. Okay. Good evening, all. So today we are scopolamine induced cognitive impairment model in adult zebrafish, and simultaneously we are perform the another experiment later we will, that is the uh, antidepressant activity by using the tail immersion tail suspension model, and third one, that is the we are performing the EPM. So very short time period of time, so all these experiments we have to perform here, but we will clarify one by one. So first we have to target, that is the zebrafish. So in that zebrafish, that is called as a Danio ririo. It is a 3 to 5 centimeter. You have to see the length of that zebrafish. That is a 3 to 5 centimeter length is occur. So body weight is around the 300 milligram to 500 milligram weight of that zebrafish. There are number of 5 zebrafish you have to take in one group. So you have to see that there is the different standard control group where we have to use the donepezil, then another rivastigmine drugs. Here there is the drug, that is a disease control group. So where disease control we have to use the only, that is the scopolamine to produce the amnesia. So this is the scopolamine which is used for the producing the amnesia, and that is prepared the 200 micromolar concentration. And third, this is the called as a feed. So that feed which is given to the zebrafish. If you see that, but before start to that, I want to clear that just like as a rat, mice, rabbit, we have to take the ethical clearance from the CCSA or IAC. So similarly here also IAC you have to take the permission to perform the experiments. So there are so many experiments we have to perform in the zebrafish. So first, that is Alzheimer models. Another model, diabetes, you have to also perform in the zebrafish. Then PCOD also we can perform in the zebrafish. So we have to handle the carefully. We have to first see the basic concept regarding about the. So in the housing and maintenance, so this is called as a glass aquaria. In the glass aquaria, you have to see, just look at this glass aquaria, where we can place the zebrafish. So in that zebrafish, it is maintain stock, that is a 5 fish in the 3-liter waters, and maintain the temperature, that is a 26 degrees Celsius by using, you have to check by the thermometer. The pH should be of this water, that should be around the 6.6 to 7.7, and similar to the rat and rabbit, rat, mice, you have to use the light cycle, that is a 12 hours. So here you have to provide the feed to the zebrafish, that is a commercially available feed into the aquarium, and whatever the water, that is the most important, that is water you have to replace in alternative days, but you have to replace the half water from the aquarium and place the new water, because whatever the good bacteria are required, that should be helpful to the survival of the zebrafish in that condition. Next important things here, that is the acclimatization. So similarly to the in CNS model, in the rat, mice, rabbit, we have to provide the acclimatization areas. So here also is the 10 days acclimatization is required for the any experiments. So that can be helpful to reduce the stress in the animal model. Next, that the chemical reagents are required. As I told, there is the scopolamine is used here, that we have to prepare the 200 micromolar concentration. Rivastigmine, that is the standard drugs that we have to use here. The test drugs, whatever your test drug, that is your synthetic compounds, maybe extracts, maybe phytochemicals, maybe isolated compound, maybe that can be used here. Second important things, scopolamine. Scopolamine we have to given to the animals, not by only, just immersion methods. Immersion method means take a beakers, we have to fill up with the aquarium, put the zebrafish here, and just that should be prepared by the 200 micromolar. Okay, so in these areas where we have this scopolamine, and that animals are producing amnesia. Regarding the test compounds, that test compound we can provide by oral, but there is a requirement, that is a micropipette. So in that micropipette is around 1 to 20 microliter. So whatever your dose, just like in the animals have to dose, there's a 200 milligram, 400 milligram, but here whatever your dose is 5 mg, 1 mg, that dose should be paired in the 1 to 20 microliter. And by using the micropipette, you have to given the dose to the animals. Okay, so request stopwatch purpose. Now specification for the T shape, maze. So there is a different books have different measurements have given. So here it is we calculate as a long arm. So this is the long arm. You have seen that the 44 length is there. Then there is the 7th centimeter is a width is there, and height is around the 10 centimeter. Okay. There are two short arms. So here you have to see there is a two short arm. So there is the length is a 17 centimeters, 17, 10 height, and 13 centimeter like this, here the length. And one is a start box. So you have to, if you want to prepare the such instrument, so you have to prepare the 3 different aspects and then attach all the 6. So this is called as a box, long arm, and this is the 2, there's a short arms. Okay, and water depth should be around the 6 centimeters. Okay, and whatever the temperature of this water is around the 28 degree Celsius. Now, if you see in this model, here is start box, long arm, and 2 short. So here are 2, there are different colors you are going to object. One is the green color and second is the red color. After specification, next that is the groups. Similarly, in the mice rabbit, we have prepared the different groups that you control, positive control, disease control, and test control. So negative control where there is we are not given any kind of here. So here also there is a control group is there. So we are providing only CMC. CMC carboxymethyl cellulose, for which purpose just only dissolve your drug sample. Second, that is a dissolve, that is scopolamine. So here scopolamine is prepared for micro, and that solution you have to prepare in the beaker around the 200 ml, 100 ml, but you have to prepare the concentration that is a 200 molar. Standard group that is the rivastigmine 1.5 milligram, along with the disease control that is a scopolamine, and whatever your drug sample that you have to prepare, but only is given by the oral route. Now dose preparation is carried out. In the dose preparation, only most important that is what are the 10 milligram, 20 milligram, 100 milligram. Synthetic compound should be less, that is a 10 to 50 milligram, and herbal that is more than 100 milligram. But you have to prepare in the micromolar quantity. Okay, and here the most important thing there is a gentle handling. Gentle handling, please, zebrafish. Just I will show you how to handle animal by using the see by using the net. Just a minute. Okay, now right you have to this animal is place into the start box. Okay, so I have put this animal from 2 days in for the acclimatization. Okay, so I have here make the doors, first door, second doors, third and fourth doors. Okay, so for first training purpose, here we are into the behavioral model. Let us see experimental time. So in the experimental time, first day 1 to 10 for the acclimatization of the animals. For first, okay, allow each fish to explore the maze for 4 minutes per session and conduct one session per day for 6 days. So 1 day, 1 session, and that session you have to continue for the 6 days. But here where the explore, that is the 4 minutes into the one areas. Here also you have to provide the 4 minutes food experiment. Okay, when we are going to perform the experiment on the 7th day, before day you have to fast the animals, do not provide any kind of the foods overnight. Okay, then administered the standard drug rivastigmine, your test drugs 2 hours before the scopolamine exposure. Okay, once you have to provide the test drugs and standard drug, then these animal whatever treatment is given standard and test drug that are exposed to scopolamine, 200 molar scopolamine solution in tank for before evaluation. Okay, so your standard drug start to work in the presence of the scopolamine, your test drug also start in the presence of the scopolamine, and control group receives only vehicles. Okay, after 1 hour, next you are start the behavioral test, that is the place the fish into the start box for a 1 minute and just check within 1 minute the fish which is a from the long arm and enter into the 2 box arm, where if you see that there is the scopolamine induced amnesia, the fish which is a travel in between there, not able to recognize where to go. The treated, drug treated zebrafish which is enter into the they enter into the green zone, and that you have to count the entries, total number of entries in each short arm, or how much time spent by the zebrafish into the green arm, that you have to focus it. So here your target is that is the, to focus the 2 points, that is the number of entries and time spent in the green zone, and that is your assessment of the memory of the zebrafish. Okay, and whatever the data get, that is the number of entry and the time, that data you have to put into the Excel sheet and calculate the whatever results which is obtained by your drug sample in the zebrafish. I hope all of you have clear the concept. It is a just T-shape apparatus and where we can perform the models. Okay. So here we stop about the zebrafish. We go for the next model, that is the elevated plasmase apparatus. Okay. No, no, हे ठेवा मॅडम. आता फक्त EPM घ्या, 5 मिनिटात संपू. Hello. Okay. So good evening all. So again we are after completion of the zebrafish demonstration. So we require the ethical clearance for the zebrafish, and for that purpose in one group at least you have to choose the 5 animals for the zebrafish studies. So very interesting model is there where keen observation, silence, ambience, environment, all these parameters should be required for the zebrafish studies. So similarly, we are going to perform the another model, that is the elevated plus maze model. So all of you know in the morning session, Dr. C.R. Patil has explained the details about the elevated plus maze model for the anxiety purposes, where we can check the anxiety activity, anti-anxiety activity in the mice. So here we are prepare the 2 groups in which 3 mice are there that are control, and another 3 mice are there where are the drug treated mice. So I'm not going to explain the details about the elevated plasmas. All of you know very well all these things, but I just want to say that many colleges have pharmacology department and they can prepare this elevated plasmas model in that college level. But as we think about that, this model is only used for the anxiolytic study, that is the anti-anxiety study. But rather than apart from that anti-anxiety study, around 6 to 7 another experiments we can perform on this model. But here we need the more clear what we say, that is a clear concept about these models. So just I want to here say that the Elevated plus means there are two open arms are there, and this is the two closed arms are there. So this model is only specially for mice purposes. Rat model is different are there. So here the open arms around the 30 centimeter and 5 centimeters are there. In closed arms, similarly 30 centimeter long and 5 centimeters is the width, and 15 centimeter is the heights are there, and central platform is the 5 into 5 centimeters are there. Okay. And here the sir told about the dim light illumination is more important to perform the no any noise, any kind of disturbances required for this elevated place apparatus. Only white where just camera is required to get the report of this elevated maze apparatus. Now we are going to see that which experiments we are going to perform in these models. Let's see first that is the anxiolytic, that is anti-anxiety drugs, where the two arms and closed arms are there, and these elevated plus maze height is around the 50 centimeter, where we are just see that the animals are normally feel the fear to travel in the open arm. Okay, and whole study is mostly carried out for 5-minute duration. Okay, so here in the center, animals should be face to the open arm, that's most important, face to the open arm, and check the increased time spent in open arms, increased time spent in the open arms, increase number of entries into the arm, okay, and decrease the time in closed arm. So then and then we can say whatever your drug sample that is showing the anti-anxiety activity. Okay, so due to the presence of our voice, noise, so animal may be or may not be shown the results, but it requires the quiet. So, but placing of the animals at the center for the anti-anxiety activity, and the face should be towards the open arms. Okay, so here we have to use the diazepam drug, alprazolam drugs. So we have given the diazepam drug dose is of 5 mg per kg to the animals, and we have to place the animals in the center part. Now, if your drug has produces angiogenic activity, it produces the anxiety, that can be also checked here, where the animal can be placed more into the closed arm. Okay, then and there we can say that your drug has produces the anxiety. Such an example that is all up in very well, PTZ. PTZ is the pentylene tetrazole, which is used for the anticonvulsant activity, is related to like petimal, not for granule. ME is induced for the granule and petimal for that for PTZ. So PTZ, it is acts as angiogenic. So any kind of drug under sample that can be produces angiogenic, that we can also check in this model. Second, CNS depressant. All of you know very well CNS depressant we have to see immediately in front of the actophotometer. Okay, sedative hypnotic, its produces the sleep. So that can be model, that can be study also performed on this model. Here no change in the apparatus, same apparatus is there. Only observation, that is the locomotor suppression. In actophotometer also we have to check the locomotor suppression. Here also we have to check the locomotor suppression. But here another open field model that can be also used along with that model, and decrease the number of arm increase in open as well as in closed arm. That is the most important parameter here. Also decrease the exploratory behavior. So animal is a maximum time it is a present in the closed arm, and their movement in the closed arm also decreases. That decreased locomotor activity, even reduced rearing and head ping. That is also one of the parameters that we can helpful to determine the animal have a CNS depressant activity. Okay, if open arm time decreases, animals have open arm time decreases with a reduced total entries, they called as sedation, not a anxiety. Okay, clear? This is the second model, CNS depressant sedative models we can check into this model. Third model in the EPM, third experiment in the EPM, we have to check that is a cognitive function. Even it is called as a anxiety related memory. Anxiety related memory in which the face should be center and the animal should be placed on open arm end. Animal should be placed on the open arm end. Face should be like as the center. So here we are check the learning memory, that is emotional memory we have to check here. So here we have to give the trial, repeated trial of EPM or transfer latency is important. Okay, so first day is the acquisition trial for the animal, and second day we have to check how time required to recall the memory from the travel from the end to the another end. So here our target is a transfer latency in the cognitive memory. Here target is a transfer latency. What is the transfer latency? Time taken to move from one arm to the closed arm. Here or here. Time taken to move from one arm to closed arm. So decrease transfer latency on day 2, that indicates improved memory, and increase transfer latency that will lead to memory impairment. So this EPM model can be used into Alzheimer's disease or dementia models. Okay. New topic. So new topic model is there. So place the same animal here open arm and face it to the center and perform the same activity as the cognitive memory. Okay. Third model we have to check, that is the motor coordination, where the rotor are used here. So similarly here we have to check, place the animal to the center and facing to the open arm. Place the animal center into the open arm. Okay. It is a just only observation based motor coordination. But here you have to see only the freezing behavior. Animal should be stopped at the side, freezing. Poor arm exploration, animal is not able to poor arm. Okay, falling or imbalance. Okay, motor coordination and reduced arm transition. So these parameters we will cover under motor coordination. And last model is the stress-induced behavioral changes. So here also called as the adaptogenic activity. So here mobilization stress can be given, port stress can be given externally, restraint, place animal in the holder, animal holder, give the restraint stress, and then again keep on this active photometer, and increases closed arm increase time and test drug opposite, that is a stress induced anxiety. Only that is the pattern we have carried out in this model. And last model, that is a withdrawal anxiety. Here we have to chronic administered to the ethanol to the animal, okay, benzodiazepine chronic administration, and sudden stop that drugs, and then check how much time is required. The animal placed into the closed arm, decrease open arm exploration, and reversible, that indicates you has shown the activity. So this is the small model, very nice model, elevated plus upright model, where we can perform the various activities on the elevated plus model. Not only anxiolytics, more than anxiolytics, 4 to 5 activities we have from the EPM. So thank you very much. You have the information about the elevated plus model to get the information about the various activities. Okay. Hello? Okay, now we are going to check the one more model, that is a tail suspension model, which is used for the antidepressant activity. Okay, so in that activity, it is very nice activity, antidepressant model, where we are using the various standard drug, that is the fluoxetine, imipramine drugs we are given to the animals, and we have to check the antidepressant activity. But here in these studies we have to see that there is the animals which is hang on the uh, tail. So here this tail suspension model, the most important task is that is the animal should be hang in the air for the 6 minutes. Okay, within these 6 minutes we have to provide the such a platform where we cannot get the any kind of the platformal replaceable. So if the animal is touched to the anywhere, so he didn't show any kind of the action. But once it is clear as that, so animal is not able to escapable, animal is not able to go anywhere, so now he goes into the immobility phase. So in the immobility phase, animal should be just stable on the one phase. Okay, and whatever the animal antidepressant drugs are the different concentration that are given to the mice now, and mice has a start to the immobility, and whatever the duration is occurs for the immobility, that we have to measure in the antidepressant. Most of the colleges I have seen that to perform such a models in the mice, they have placed the animal in inescapable form for the 6 minutes and just write down the duration of the hanging of the mice on the bar. Okay, and how much time is required to reduction of this their mobility, they have changed. So antidepressant drug decrease the duration of immobility, decrease the duration of immobility. Only that point is required here. But sometimes some Scopus journals, they want proof about that. But just I have mentioned on that there is only just 6 minutes are there, 5 minutes are there, and our animal has shown the activity. So in every college have the Sherrington rotating drum machine. So that Sherrington rotating drum machine you have to place here. Okay, animals should be, so this is the Sherrington rotating drum machine. And just remove this tail suspension apparatus sidewise and place the student organ bath. I will just show you the how we have to prepare. Okay, so this is the normal assembly we have to perform in our colleges. Now here same say animal, that is a mice, that is a suspend here at that site. Okay, and you have to just apply the tip. So whatever the inescapable form, so animal shows immobile. Okay, so there is just a single line is occurs. Okay, an antidepressant which decreases the duration of immobility, so the animal start to move the movement. So here, see here, the already we have to perform the same experiment, and here the animal has shows the fluoxetine drugs as given to the animals, and that have shown the such type of results. So you have to cut this and put into the your paper and send to the as a graph a normal animal reading and fluoxetine drug given to the animal. So this can be differentiate clearly the animal, your drug sample has shows the antidepressant activity by using the tail suspension model. So this is also one of the model I have learned from the, I have visited to the Punjab University in the laboratory of Dr. S.K. Kulkarni. So I asked about this model, so that at that time they have shown the Sherrington rotating drum machine model where we can take the proofs, and nowadays every journals have want to the proofs regarding about their activities and all these things. So this is also one of the manual methods that can be converted into the reading methods on the Sherrington rotating drum machine. So this is also one of the best model for the antidepressant that we have to perform in the our laboratories. So thank you very much for showing such an antidepressant model by using the rat and mice on the Sherrington rotating drum machines. Okay. Okay, thank you very much to all of you to listen our sessions. In this session, we have explaining about the zebrafish model. So where we can perform the various activities on the zebrafish, like as the Alzheimer diabetes, then PCOD. There are so many models are there, CNS, CVS, GI Tech. But carefully handle all the animals, do not stress to the animals, and enjoy the experiments with their proper results. So similarly on the rat mice also we have to perform the different activities that can be also performed the very well manner, and animal will definitely show the result of your drugs if you perform the honestly, systematically, properly with the guidance and properly with the protocols. So thank you very much.
Red blood cells, white blood cells, and platelets. These blood cells are essential for carrying oxygen to our organs, fighting infections, and helping our blood clot. Types of bone marrow. There are two types. And what does it do? Bone marrow is a soft,
Check one, check. Hello, hello.
बॅकग्राऊंडला
Good evening, everyone. Now, in this last demo session, we will be performing iliac bone marrow aspiration in rabbits. The main surgery will be performed by Dr. Santosh Pika, and he will be assisted by Dr. Suruchi Pawar, Associate Professor, Department of OBGYN. Also assisted by Sonal Patil, 4th year PhD students at Stem Cell and Regenerative Medicine, and Harshada Lokhande, 2nd year PhD student at Stem Cell and Regenerative Medicine. Also animal house handling expert here, Akshay Thoraskar, and Afroz. Before starting, I would like to show you the demonstration, a video demonstration of iliac bone marrow aspiration in rabbits.
And what does it do? Bone marrow is a soft, spongy tissue found inside our bones. It is responsible for the production of all blood cells in our body, including red blood cells, white blood cells, and platelets. These blood cells are essential for carrying oxygen to our organs, fighting infections, and helping our blood clot. Types of bone marrow. There are two types of bone marrow, red bone marrow and yellow bone marrow. Red bone marrow is found in the flat bones of our body, such as the pelvis, skull, ribs, and sternum. It is responsible for producing all types of blood cells. Yellow bone marrow, on the other hand, is found in the long bones of our body, such as the femur and humerus. It stores fat and does not produce blood cells. Blood cell production. The process of blood cell production.
This was a small demo at the bone marrow aspiration. Now we will move, I'm sure you can see the surgical site on your display. Now, why do we perform this bone marrow? What are the different applications of this aspired bone marrow? The major and the most primitive use of this bone marrow is for regenerative medicine. These bone marrow cells contain stem cells, which are mesenchymal stem cells, hemopoietic stem cells, which are used for different application in regenerative medicines, that can be used for endometrial regeneration, regeneration for different lower limb diseases and disorders. Here we see what is the bone marrow and what does it do. Now, when this bone marrow would be aspirated, there are different type of cells. Now, when one asks, what is the bone marrow and what are the constituents? You can see that RBCs, platelets, WBCs are the major red bone marrow cells. In the previous video, you already saw there are two types of bone marrows, red bone marrow and yellow bone marrow. In red bone marrow, you can see these three types of blood cells. For performing this iliac crest aspiration, the preoperative preparation of the rabbit for iliac bone marrow aspiration have been done. Minimum 6 to 8 hours ambient before anesthesia. That is, you would make sure that the rabbit you're going to use for a surgical site is kept under fasting. There is a surgical instrument preparation. You make sure all your general surgical instrument have been autoclaved, sterilized completely. And then the major important step for the rabbit, that the New Zealand rabbit that we use, that is pre-anesthetic and anesthesia, dosage, xylazine and ketamine. Ketamine would be given as 35 to 40 mg per kg, and xylazine would be given in the dose of 3 mg per kg. The rabbit that we are, there's a animal that model that I've been using right now is up to 2 to 2 kg. So the dosage would be 60 to 65 mg per kg for ketamine and 6 to 7 mg per kg for xylazine. Now we would go for a preparative of a surgical site. Now here you can see the surgical site have already been prepared and they're giving an incision.
We have hello, am I audible? We have palpated the spine and we are going lateral to the spine to palpate the iliac crest. And here it is the site of the incision, which will be around 2 centimeters. I repeat, we have palpated the spine of the rabbit and we are going lateral where we are able to palpate the iliac crest. And here is the incision that will be taking around 1 to 2 centimeters of length.
The rabbit was under anesthesia. The anesthesia was given to the rabbit 10 to 15 minutes before the surgical site was prepared. As we previously discussed, 60 to 65 mg per kg of ketamine and 7 mg per kg of xylazine, muscle relaxant. Continuous checking of breathing. If the animal that has been put under surgery, that is, if it's continuously breathing, we need to check if their heart rate is increasing or not. these different behaviors are need to kept under check.
The layers that we have incised is the skin, the subcutaneous tissue, the muscle. And now we will be trying to go to the bone, that is the iliac crest.
Now one would ask, why iliac crest and not femur or any other bone? Now the iliac crest is chosen because the cells are abundant. The MSC stem cells that are present in the iliac crest region of the pubic bone of the hip bone of the rabbit. Hence, this region has been chosen over a femur or any limb bone aspiration. First we go to skin, then muscle, and the final periosteum layer of the bone. One might ask, who have no idea about aspiration, what is and why is a bone marrow aspiration? Because this technique is autologous and minimally invasive. It's the minimally invasive source of bone marrow. It is clinically most relevant and, of course, ethically acceptable widely. You make sure that the least area is exposed to the surgical site so that there are no chances of infection while we are performing the surgery. I previously discussed that the major application of this bone marrow, which has been aspirated from rabbit, is used for endometrial regeneration or regeneration of endometrial lining. Now, why would we use a rabbit model for this? Is because the rabbit's uterus closely mimics the human endometrial repair mechanism. Also, the suitable size for uterine injury and intrauterine cell delivery. That is what helps us establishes this rabbit model for preclinical reproductive model. Here you can see a clear incision of the needle into the iliac crest region.
we have tried to negotiate the periosteum of the iliac crest.
There are different size gauze that have been used for aspiration. The biopsy needle and the 18 gauze size needle. Based on the different size, there are different gauze needles that have been used. For rabbit, we use 18 gauze needle. You can, while performing the surgery, while inserting the needle, you can feel the iliac crest going completely inserted into the region. You make sure the correct angle of the needle so that the bone marrow can easily aspirate. If you understand the basic gravity, you can tell that the bone marrow would be directed and would be taken by a syringe. Negative pressure. Would be applied to syringe to aspirate the bone marrow. Meanwhile, the heart rate of the rabbit is normal. Currently, one ongoing study has been going on under the Suruchi ma'am, Suruchi Pawar ma'am, assisted associate professor at OBGYN. Her PhD work is currently on Asherman syndrome, which can be regenerated the lining of endometrium by using bone marrow MSCs. Red bone marrow serves as the primary factory for blood cell production because it contains the body's, as I previously said, hemopoietic stem cells. It's the essential target for diagnosing blood disorders and vital resource for life-saving treatments. There's so many diagnostic applications of these bone marrow stem cells. It can be used for diagnosing cancers. It's the definitive method for identifying the blood-related cancers like leukemia, lymphoma, and multiple myeloma. Staging and monitoring. Investigating the blood disorders, identifying different conditions such as iron deficiency and even source of persistent and unexplained fevers. There are different treatment applications. Treatment involves utilizing stem cells within red bone marrow to restore a patient's ability to produce healthy blood. That can be done by bone marrow transplantation. This rescues the blood after high-dose chemotherapy or radiation that destroys the diseased marrow. It is the cornerstone treatment for the leukemia and severe aplastic anemia. There can be different resources such as autologous, allogenic. Autologous, that means uses the patient's own healthy stem cells, often harvested and frozen before intense cancer treatment. Allogenic, which are used cells from matching donor, often sibling or unrelated volunteer from registered at NMDP. Gene and immune therapies. Modern treatments like CAR T cell therapy use genetically modified cells, often derived from marrow precursors, to specifically target and kill the cancer cells. And the last one for treating the genetic diseases. Bone marrow transplantation can replace malfunctioning marrow in children with the inherited conditions like sickle cell disease, thalassemia, or metabolic disorders like Hüller's syndrome. So overall, you can see that the bone marrow had a wide range of treatment applications, from cancers to sickle cell disease to genetic disorders. Using autologous and allogenic ways, we can help cure these patients. There are different animals used for bone marrow aspirations, from small animals such as rat, mice, to larger animals like rabbits, guinea pig, chicken. From small animals, we can acquire a very few drops of bone marrow, as they are smaller in size and the production of bone marrow is little as compared to the smaller ones. Here we can see you can successfully insert it into the iliac crest region of a bone marrow by using 18 gauge needle. Now you can see the 18 gauge needle is being inserted after removal of the rod. One might say it looks easy, but it is a bit difficult and tricky to locate the iliac crest region and to successfully insert it without harming the surrounding area. Here you can see the set of general surgical instrument and the needle that we described. First step would be the incision and iliac crest exposure for bone marrow aspiration.